Welcome all in this special session of today. This is the second time that we organize a forum on immunotherapy within the context of this very important meeting dedicated to malignant lymphomas. Thanks to the active collaboration with Professor Franco Cavalli and Professor Andrea Limonti of the Institute of Oncology Research in Bellinzona here in Ticino. I take this opportunity to thank Professor Cavalli for the great work he has done for years and also for his special commitment to the people. The IPSA Foundation was founded by the pharmaceutical company IPSA in 2012 with the aim of making scientific dissemination because we believe it is our duty to transfer scientific knowledge to the population, therefore to everyone. Scientific culture is very important because it will increasingly influence political choices and decisions that are crucial for our future. We organize forums like this today to make networks, of course, and to encourage the sharing of important research like those that will listen this afternoon. But above all, because we want to bring the correct scientific information out of the laboratories and give it to everyone. It is absurd to think that those who create fake news have greater power doing damage and causing real problems for everyone without even realizing it. While professors are often only talking to each other, the scientists who will give talks today have dedicated their lives to research, have achieved important results for all of us, and are also great speakers. Our foundation is committed every day in this direction with fellowships activities, forums, workshops on frontier science topics, labs in the school, and much more. And I invite you to follow us through our digital channels, to which we devote a lot of efforts. I'd like to thank the people who work with me every day and believe in this very important project. Before giving the word to Professor Andrea Limonti, who will introduce the forum and will be the chairman with Professor Cavalli of the afternoon, I remind you that out there is a coffee station with soft drinks out from here. We remain all the time for you. So before starting the meeting, I call Professor Cavalli for a welcome from you.
Thank you, Dr. Miziti. Welcome to everybody. Good afternoon. It is a pleasure for us, for the Lymphoma Conference, to have for the second time this special workshop organized by the Foundation IPSA. As you have seen, there will be 4 very prestigious speakers who will discuss with you revolutionary treatment in oncology, in immuno-oncology, mainly today. We are also very glad because for the second time, the person who was awarded the San Salvatore Prize and held the Henry Kaplan Memorial Lecture, which is the most important keynote lectures of our Congress. Dr. June will be one of the 4 speakers. The Lymphoma Conference is going very well. We have almost 4,000 participants. At this very moment, we have 4 concurrent sessions. And just close to here in the hall of the university, there is a joint meeting and a joint session organized by our conference together with the Chinese Union of Lymphoma Researchers, investigators, and you might understand that for diplomatic reasons, I will have to go there, and so I will be only very partially here. But I am sure that this afternoon is going to be very interesting for you, and thank you for being here.
I think that everybody knows Professor Cavalli, and so now I introduce Professor Andrea Limonti, that is younger, so I have to say something about him. He's a professor here in Lugano at the University of Italian Switzerland, has twice received the ERC grant, and is currently head of the Molecular Oncology Lab at the Oncology Research Institute in Bellinzona. Author of several publications in the most prestigious scientific journals, and in addition, is also member of our Foundation Scientific Committee. So please, Professor Andrea Limonti.
Well, thank you very much. Thank you, Silvia. Thank you, everybody, for being here. I prepared another introduction, but perhaps it's better don't do it because we have done many introductions. But I just want to say to the people that is in the audience is that this meeting is supported by IPSA Foundation, is open not only to the member of ICML, but also to student, postdoctoral research fellow, young clinician of our Faculty of Medicine, and that are part also of our affiliated institute. So I think that this fourth speaker of today, they give a gift not only to the ICML, but also to the students that are in the audience. So today I have the honor of to open this meeting, which we will see four amazing scientists which have done incredible contribution in cancer research. Not only a basic contribution, but also contribution in moving this discovery to the clinic. And if you have the patience to stay with us from the beginning to the end, you will learn, for instance, from Michael Hall about mTOR, and how we can target mTOR for cancer therapy. We will later on have the pleasure to host Alberto Mantovani. We will discuss with Alberto about the role in innate immunity in cancer, about inflammation, and now also to target population of cells which have been so far neglected, I would say, by pharmaceutical companies. So these are neutrophil and tumor-associated macrophages. After, we will have the great privilege to host Carl June. So Carl is the father of the CAR T-cell therapy, and we are really honored to have you here today. And finally, we are also pleased to have the president, the elected president of ESMO here, so the president of all of us. Actually, today we are lucky because we have two presidents, the president of ESMO and the ACR, which kindly agreed to came to the meeting. So with this, I start the meeting, and saying that, I want to introduce Michael Hall. Michael doesn't need an introduction. He's a professor in Basel, and he received a super prestigious award in 2017, which is the Lasker Award, for the original discovery on Tor. So, Michael, we're really happy that you made it here, and that you will give us some insight about Tor biology and therapy.
So, good afternoon, and I'd like to start by saying it's a huge pleasure to be in the enchanting Ticino, and in particular at this IPSA Foundation sponsored forum. So many thank you to the organizers, Andrea and Franco and Silvia, for bringing me here to Ticino. So we work on cell growth, and in particular work on the Tor signaling pathway. And what I'd like to do today is tell you one story, well, two stories perhaps, depending on how much time I have. Both stories can be bundled under the general theme of mTOR signaling, growth, and metabolism. Both stories have to do with cancer, how mTOR is involved in cancer, in this case in particular in the liver. And the first one is how Tor-controlled histidine phosphorylation is oncogenic, and then the second story, if I get to it, will be how mTOR controls HCC via controlling lipogenic biosynthetic pathways. Before I tell you these stories, I'd like to give you a brief introduction to Tor signaling. So Tor is, and here it's shown in its generic form, Tor. Many are familiar with mTOR, which stands for mammalian Tor, but Tor is conserved all the way from yeast to human, and so we often refer to it as Tor as in the generic form. So Tor is a kinase. It's a serine threonine protein kinase, highly conserved, as I just mentioned, and it is also known as a central controller of cell growth by virtue of the fact that it controls a large number of cellular processes which collectively determine mass accumulation in the cell and thereby control cell growth or cell size. And these processes can be subdivided into two groups: the anabolic processes which Tor activates and the catabolic processes which Tor inhibits. So Tor balances these opposing forces of synthesis and degradation and does it, and this is important, does it in response to nutrients. So in very simple terms, Tor is what makes us grow when we eat. And of course, this is then what's led to this notion that Tor is a central controller of cell growth. So how does Tor actually control all these processes? Well, signaling pathways emanate from Tor to intersect with key proteins involved in every one of these processes. And in some cases, we know what those processes are, but in many cases, we do not yet know. But this is an overview of what I call the Tor Signaling Network, and I call it a network because it's more than a single pathway. It's in fact two pathways, each defined by a Tor complex. So Tor is in fact two structurally and functionally distinct kinases, each one of which phosphorylates its own substrates to control its own cellular processes. Now, like Tor itself, these two complexes are highly conserved all the way from yeast to mammal. Indeed, the whole network is highly conserved. So the picture that's emerged over the last 10 to 20 years is that this is an ancestral or primordial signaling network which has been conserved throughout eukaryotic evolution to control this very fundamental process of cell growth. Now, the sole exception to that statement I just made is this part of the network up here, which is the growth factor or insulin signaling pathway, which evolved much later. This evolved with multicellularity and was then grafted onto this more ancestral Tor branch, which already existed in unicellular organisms. And the reason for that is that cell growth in metazoans or multicellular organisms is more complex than in unicellular organisms, because in metazoans it's critical to control the growth of every cell in the body with every other cell in the body. So you need an input which controls growth over a whole body plan to ensure that our organs are all properly proportioned. And this then is what's provided by the growth factor signaling pathway, and that's why this then evolved much later, evolved with multicellularity. Now we know in mammals or in metazoans, we have the growth factor input, nutrients, as already exists in yeast, and in this case, amino acids are the key nutrient. Leucine is a particularly good nutrient, and now we also know that energy in the form of ATP is required to upregulate the Tor signaling network. So these are the three inputs which activate the Tor network: growth factor, nutrients, and energy. And once these three factors are present, the network is upregulated, all these anabolic processes are activated, and cell growth ensues. Now, another rather fascinating aspect of the Tor signaling network is the unusually large number of diseases to which it's been functionally linked, mainly genetically. And what all these diseases have in common is they're characterized by ectopic or inappropriate cell growth, again underscoring this notion that Tor is a central controller of cell growth. And this can be either malignant or benign forms of cell growth, but in all cases, cell growth. More recently, Tor has been implicated in another set of disorders, the so-called metabolic disorders. We know that chronically high circulating levels of nutrients can upregulate Tor even in the absence of growth factor, and this can then lead to adipogenesis and in turn obesity and ultimately type 2 diabetes. But we have more direct links to type 2 diabetes, more direct links between nutrients and type 2 diabetes via this famous negative feedback loop in the Tor network, where Tor or S6 kinase just below Tor phosphorylates IRS in the signaling pathway to inhibit it, and this then confers insulin insensitivity, which, as you know, is one of the hallmarks of diabetes. So in this context, some have actually proposed rapamycin as an anti-diabetic because if you inhibited Tor, you'd inhibit the negative feedback loop and you'd restore signaling through the insulin signaling pathway. I don't believe any pharma company is actually developing rapamycin as an anti-diabetic because there are a number of complications which would argue against that, but something which gives credence to this notion is the fact that metformin, the world's most commonly prescribed anti-diabetic, works at least in part by downregulating Tor via activation of AMPK. So Tor is involved in many disorders, so you would suspect that rapamycin, and Tor is an acronym which stands for targeted rapamycin. Rapamycin is a small natural molecule which binds and inhibits Tor. You'd expect that given that Tor is involved in so many diseases that a drug which binds it would be used in the clinic, and this is indeed the case. This is rapamycin, this natural product secreted by a soil bacterium first found in Rapa Nui, hence the name rapamycin. And this molecule, despite its very humble beginnings in the dirt in this very faraway place, is now used in 3 major therapeutic areas, originally immunosuppression, now more famous as an anti-cancer drug, and it's also used in the treatment of cardiovascular disease, restenosis after angioplasty in particular. And just to give you a feel for how recent some of these developments are, here's a timeline of these clinical applications of rapamycin. It was approved for immunosuppression 20 years ago this year. About 15 years ago, it was used for treatment of restenosis. And then about 10 years ago, Pfizer had a version on the market by the name of Toricel for the treatment of renal cell carcinoma. And Novartis more recently has a version called Affinity Tor, which is now used for a number of different cancers. So it's a drug which has had remarkable applications in very, very different therapeutic areas. So while we and others have been trying to understand the physiology and pathophysiology of the Tor signaling network, we've also been trying to understand Tor at the atomic level. And this is the structure of human Tor complex 1, which we determined a couple years ago by combining X-ray crystallography and cryo-EM. Tor is a dimer, a homodimer of heterotrimers. It has 2 catalytic sites, one here and one on this side. We also determined the structure of Tor complex 1 when it's bound to its inhibitor, rapamycin. And what's important to realize that rapamycin doesn't bind and inhibit Tor by itself. The mechanism of action requires that rapamycin first form a complex with another small protein called FKBP, and this FKBP rapamycin complex then binds directly to Tor. And for many years, we wondered why rapamycin alone could not inhibit Tor complex 1. Why does it need to form a complex with this protein? And we determined the structure. We understood the mechanism. So this is FKBP, and rapamycin, if you could see it, would be a sort of glue which glues FKBP to Tor in this case. And as you can see, there are 2 FKBP rapamycins bound to each of these 2 binding sites in Tor complex 1. And as you can see, the mechanism by which rapamycin inhibits, or FKBP rapamycin inhibits, is it binds the lip of the catalytic cleft, thereby excluding access of substrates to the catalytic pocket. And this then explains why rapamycin alone does not inhibit, because it's simply not bulky enough to form this sort of a lid which covers the catalytic pocket. It took us several years to determine the structure of Tor complex 1, about 10 years. And the reason is that it is a rather large molecule. You can't appreciate it from these images, so let me give you a size standard which allows you to put this into context. So here's Tor complex 1, standing it up, and here's something which we all know, the ribosome. So Tor complex 1 is about the size of the ribosome, which is much larger than I thought it would be when we first discovered it decades ago. What should also strike you is that the ribosome is a much more dense structure than Tor complex 1, And I think this actually reflects how little we still know about Tor complex 1. Once we know the structure of Tor complex with all its regulatory proteins bound, it might look like something approximating the ribosome. So we still have a great deal of work to do. Even more recently, we discovered we elucidated the structure of Tor complex 2. It is also a dimer, now a dimer of heterotetramers. Tor itself in the two complexes has the same structure, but it now binds different subunits to create these two structurally and functionally distinct complexes. Determining the structure of Tor complex 2 also solved a puzzle which we'd been dealing with for many, many years, and that's the fact, actually since 2004, the fact that Tor complex 2 is insensitive to rapamycin. So why could rapamycin bind Tor complex 2, inhibit it, or rather Tor complex 1, inhibit it, but do nothing to Tor complex 2? Well, once we determined the structure, we found that one of the subunits of Tor complex 2 masks the FKBP rapamycin binding site in Tor, such that FKBP rapamycin cannot touch Tor complex 2. So this is also a rather important advance. So this is my introduction to Tor signaling. Let me now tell you the story, the first story, and the only story I will be able to tell you. And this deals with the role of Tor in hepatocellular carcinoma. And what initiated this study was the fact that it's long been known that Tor complex, mTOR complex 1 or 2, promotes cancer, but the effector pathways via which it promotes cancer were unknown. So what we want to do is determine what these effector pathways might be, and to determine that, we created an mTOR-driven mouse model for HCC. And the way we did that was by knocking out two tumor suppressors in the mTOR network, two well-known tumor suppressors. On the one hand, TSC. We knocked out TSC. This upregulates mTORC1. And then we knocked out the second tumor suppressor, P10. And we combined these two knockouts. What results is mTORC1 is even further upregulated. Now we also upregulate Tor complex 2. So this is a mouse model in which both complexes are hyperactivated. And when we produce these mice, which we call LDKO mice for liver-specific double knockout, so we did this specifically in the liver. The first thing we found was that these livers from the double knockout mice were enlarged, hepatomegaly, and this is due to the growth-promoting function of Tor, and the liver continues to grow throughout the lifetime of the mouse because mTOR is hyperactivated in all cells of the liver. Well, about 6-8 weeks of age, we started seeing fatty liver or steatosis, which then progressed to hepatitis, an inflamed liver, and by about 16 weeks of age, and knockout occurs at birth, by the way, at 16 weeks of age, we start seeing very small tumors in these livers, and then by 20 weeks of age, we have very severe HCC, many and large tumors in this liver, as you can see. We did an experiment to see whether this is in fact mTOR-driven by treating the mice with rapamycin at 11 weeks of age, and then harvesting the liver at 20 weeks. And what we found was that in the case of rapamycin treatment, we see a relatively healthy-looking liver, a little fatty, but otherwise quite healthy-looking. And as usual, in the case of control or vehicle-treated, we see this very severe HCC. So this tells us that this is a very good system in which to study HCC. Another important point is it recapitulates what's seen in the clinic, where we know that fatty liver or NAFLD is a risk factor for development of HCC, and this is precisely what we see in these mice. We see fatty liver progressing to HCC. So the questions we wanted to ask were, what are the events that give rise to these individual tumors in this liver? And are these events the same for every tumor, or are they distinct? And to answer this question, we dissected out these individual tumors and then applied a large number of omics analyses. And what I will tell you about today is purely what we found by determining the proteome of all these tumors. So we looked at 12 tumors, 3 each from 4 different mice. As a control, we pooled 6 wild-type livers. And this is when we did the proteomics, we could detect and quantify 4,000 to 5,000 proteins per tumor. And if we look at these numbers more carefully, we could in total detect almost 8,000 proteins across all 12 tumors. But if we confine our analysis to those proteins which we could detect in at least 10 of the 12 tumors, we get the number down to about 3,000. And among these 3,000, we see that 262 are downregulated and 433 are upregulated. And then to further focus our analysis, we looked at these whose expression changed in the tumor compared to the control, and looked specifically at kinases and phosphatases. And among the kinases which we detected were up in the tumors were NME1, NME2. And this caught our eye because NME1 and NME2 are the only known histidine kinases in the mammalian cell. And on the other hand, on the phosphatases, we saw one that was down, a putative phosphatase, and this is a putative phosphohistidine phosphatase. So the kinase is up and the phosphatase is down, which suggests that histidine phosphorylation is playing a role in these tumors. So let me give you a little introduction to histidine phosphorylation because it's little is known about this. It's often called the hidden phosphoproteome for reasons I will describe in a moment. Many amino acids can be phosphorylated, not just serine, threonine, and tyrosine, but also histidine. And unlike what I will call the conventional phosphorylation, serine, threonine, and tyrosine, which forms a very stable PO bond, histidine phosphorylation forms a very labile, heat and acid labile, PN bond. And this is why it's called the hidden phosphoproteome, because essentially any manipulation you do in the laboratory, any biochemical experiment you do in the laboratory to study it destroys it. So it always disappears. It also comes in 2 isomers, depending on which nitrogen in the imidazole ring of histidine is phosphorylated. If it's the nitrogen in the 1 position, it's called 1-phosphohis. Nitrogen in the 3 position, 3-phosphohis. As I already told you, NME is the only known histidine kinase. There are 2 known phosphatases for phosphohistidine, and then there's LHPP, the one we found in our proteome. Another fascinating aspect about histidine phosphorylation is the mechanism. So NME1 and 2 form homo or hetero multimers or oligomers, which then autophosphorylate on a very specific histidine, and then it transfers this phosphate to substrates either in the 1 or the 3 position. So now back to our own data. So these are the expression values for these proteins here from our 12 tumors. As you can see, NME1 and NME2 are up in all 12 tumors. The 2 known phosphatases essentially do not change. And then LHPP is down in 11 of the 12 tumors. So there was something special about LHPP. We confirmed by immunoblotting what we saw by mass spectrometry. LHPP is in fact down in the 12 tumors. NME1 and 2 are up. Next, we looked at whether loss of LHPP is specific to the tumor, and for this we dissected apart tumor and non-tumor tissue from the same livers, and we found that, in fact, LHPP is lost specifically in the tumor compared to the non-tumor. So here we have these kinase and phosphatases, suggesting that histidine phosphorylation is important for tumorigenicity. But how are we about to study this? As I told you, it's a very labile kind of phosphorylation, very difficult to study biochemically. But fortunately, at this time, I ran into Tony Hunter, who just had developed 2 monoclonals, which specifically recognized 1-phosphohis and 3-phosphohis. And these were precisely the tools which we needed at that exact moment. These reagents were like manna from heaven for us. So we immediately probed our tumors. And as you can see, there's high global 3-phosphohis in the tumors compared to controls. Very little 1-phospho-His. This is NME itself, autophosphorylated. So it appears that 3-phospho-His at least is high in the tumors. And here you can see the heat lability of this phosphorylation. If you heat the sample 95 degrees, you lose it, telling us it's indeed histidine phosphorylation. This heating would have no effect on serine, threonine, or tyrosine phosphorylation. When we reconstituted LHPP expression in cells which had lost it, cells from our tumors, we found that we could bring 3-phosphohistidine levels back down again, confirming, along with other in vitro evidence, which I'm not showing, that LHPP is indeed a phosphohistidine phosphatase. What are the functional consequences of restoring LHPP expression? Well, we find that it blocks hepatosphere formation in vitro, suggesting using this in vitro test for liver tumors that LHPP is a tumor suppressor. We did a more meaningful experiment. We injected adeno-associated virus expressing LHPP on a heterologous promoter into mice with high liver tropism. At least the virus had high liver tropism. And then we did this at 8 weeks, harvested the livers at 20 weeks. And as you can see, when we ensure LHPP expression, we suppress HCC, telling us that, again, that LHPP is a tumor suppressor. What about in humans? Well, we obtained biopsies, tumor and non-tumor biopsies from patients, and we could probe these for LHPP or NME, and what we see in the humans is the same as what we see in the mice. LHPP is lost in tumors compared to non-tumor tissue. Histidine phosphorylation is also up in the tumor in the human samples versus the non-tumor tissue, and this is just the same fact shown by immunohistochemistry. What are the clinical correlates? Well, we look at expression data from TCGA, and we find that low LHPP expression correlates with low survival, again suggesting it's a tumor suppressor. And finally, if we look in cancer genome databases, we find a number of LHPP mutations, many of which are predicted to be loss of function mutations, as you would expect if this were indeed a tumor suppressor. So we think LHPP is a novel tumor suppressor, and the extension of this, of course, is that histidine phosphorylation is oncogenic. So in the theme of the meeting, what are revolutionary therapies for cancer? I think we can start thinking about targeting histidine kinases, perhaps a novel mechanism of treating tumors. And finally, to summarize everything I've shown you, we think NME1 and 2 are controlled by mTOR, mTOR2 in this case, which leads to histidine phosphorylation. There are several phosphatases which keeps this level of histidine phosphorylation in check. But if you have a situation where you have hyperactive mTOR signaling, as you often see in tumors, at least 50% of liver tumors have high mTOR signaling. This histidine phosphorylation is still kept in check by these other phosphatases, but now on top of high mTOR signaling, you lose LHPP. You can no longer hold this histidine phosphorylation in check, and this then leads to tumor development. Of course, the key thing we have to do now is understand what are these proteins which are being histidine phosphorylated to drive promoter growth, to drive tumor growth rather. We have used those antibodies from Tony Hunter to biochemically purify histidine phosphorylated proteins from these tumors. And what we're finding is actually quite interesting. We're finding not conventional signaling pathway proteins phosphorylated, but we're finding a completely different class of a completely different type of protein that's phosphorylated. In this case, they're all metabolic enzymes involved in glycolysis, NAD synthesis, fatty acid synthesis. So I think this is probably telling us that, as we already know, that histidine phosphorylation is fundamentally different from serine, threonine, tyrosine phosphorylation, and is in fact doing something fundamentally different in cells, perhaps in this context to reprogram metabolism, cancer cell metabolism, to allow development of the tumor. Okay, I have run out of time, so I can't tell you my last story, but let me give credit to the people who have done the work. This was all the work of a very good postdoc in the lab by the name of Shravan Hindupur, and working with Marco Colombi and Dinah Weisenberger. The structural work, which I mentioned in the introduction, was in collaboration with Tim Meyer and Nenad Ban. And of course, histidine phosphorylation was in collaboration with Tony Hunter. And the human biopsies which we used were provided by our clinician collaborator, Marcus Heim, and a pathologist, Luigi Terracciano. And thank you for your attention.
So we have time for question. Alberto? Well,
of course, liver cancer is driven by inflammation. Mm-hmm. And I wonder whether when you unleash the pathways, mm-hmm. how much of the phenotype you have is dependent on a change in immunity and inflammation. And of course, you are blocking with rapamycin.
Yeah.
And
Mm-hmm. We've looked at that more indirectly. We've looked at the fatty liver that evolves, which is probably then leading to the inflammation. And we find that we've looked at the pathway, the lipogenic pathways which are upregulated. We see cardiolipin and glucosylceramide pathways are upregulated. And if we knock down these pathways, we block the inflammation, we block tumor growth. So we didn't look at inflammation directly, but we looked through the fatty acid. We have
another question?
It's a great talk. Thanks.
Mm-hmm.
So how does the hyperactivation of mTORC1 and mTORC2 lead to the loss of LHP, the tumor suppressor,
LHPP?
Yeah.
The question is, how does hyperactivation of Tor, mTOR, lead to loss of LHPP? We think these are independent events. We think that we hyperactivate mTOR signaling with knocking out our two tumor suppressors, and this makes the liver grow, and something else has to happen, individual isolated events which give rise to these discrete tumors. And we think that additional isolated event, a second hit if you will, is loss of LHPP expression. I don't think Tor has anything to do with the loss of LHPP expression. It's just the additional event that has to happen. We've looked at how we lose LHPP. We don't find any mutation in any of the tumors in the LHPP gene, so we think it's an epigenetic change in loss of LHPP expression. We know loss of transcription is the underlying mechanism.
Thank you.
I think we have time for another question. If there are, yeah. Question? Yeah.
Thank you, great talk. I was wondering if you can comment on the belief that metformin is really protective.
That metformin is a protector, an anti-cancer activity. Are you asking? I'm not sure what you're asking me. Are you asking me whether it in fact has an effect on mTOR, or do I believe the literature, or what? There is epidemiological data which suggests metformin has anti-cancer activity. We've done some experiments in the lab where we've screened for small molecules which synergize with metformin to block tumors, and we find these things. And either one alone does not work very well, but if you combine them, we do. So we do, in fact, in our lab see metformin has anti-cancer activity, and this can be boosted by co-therapy with another drug which is called serosingapine, which is a drug which used to be on the market in the 50s and 60s for hypertension. It now has an off-target effect, and we've worked out the mechanism, but this was just published in the last few months, if you're interested.
Mm-hmm. Well, I also have a question. Mm-hmm. I was curious about the fact that when you lose PTEN or TLC by themselves, you don't initiate any tumor regenesis, right? So you don't start tumor growth, right? You need to lose both of them. So did you notice whether upon loss of PTEN you have senescence induction, or you switch perhaps this senescence response by knocking down both the PTEN and TSC? This is the reason why you later on have cancer.
Well, if we knock them out individually, we do get tumors, but it takes a year or 2 years. In the case of the double, it happens rather quickly, within months.
Okay.
This is why we combine the two. We've actually never looked at the senescence issue, though. We should do that, perhaps.
So, yeah.
Thank you. Very great talk. I'm wondering, histidine is a very peculiar amino acid in terms of pK. I'm wondering if you find any correspondence in your data about the pH values of the cell and the oncogenic
No, we
activation.
haven't looked at that directly, no.
Okay, thank you.
I think it's the last question because.
So I wonder if it's a global histidine phosphorylation you believe is oncogenic, or is a specific histidine phosphorylation of a specific proteins?
It's probably something in between those two. I doubt it's a single protein, but probably several proteins, which have to be, and certainly not all of them. We don't know how many proteins are histidine phosphorylated. I would suspect there are about 1% of total phosphorylation. The protein is histidine phosphorylation, which is about a level comparable to tyrosine phosphorylation. So there are probably quite a few proteins, but I doubt all of them are involved in tumor formation.
Okay. Thank you very much, Michael. It was a great talk. So the next speaker is Alberto Mantovani from Alberto is the scientific director of Humanitas Institute in Milan and one of the father of innate immunology, innate immunity and inflammation in cancer. We discussed at lunch. He has a very interesting new story about a new phenotype that is observed in neutrophils. So I think that he's going to talk about this and about therapy that can reprogram either neutrophil or macrophage. So thank you for accepting the invitation.
Thank you, Andrea. It's a pleasure to be here. And it's a real pleasure to be invited by IPSA Foundation because of the own merit of the meeting and because of what IPSA is doing to increase the public perception of science, which I think is a key issue for us all. So my connection to the tumor microenvironment and to tumor immunology stems from my interest in macrophages, and I want to remind you of the forgotten contribution of this guy, Bob Evans, because Bob is the one who actually formally showed that leukocytes in tumors are bona fide normal cells. They are not tumor cells in these guys. And he and Peter Alexander originally showed that macrophages could kill tumor cells when appropriately activated, M1 macrophages. You pass me the definition. I had the privilege of working with Bob and Peter. My views diverged because early on, I found that macrophages isolated from human tumors or from metastatic mouse tumors did not kill tumor cells. They were actually promoting tumor growth. And actually, that led us to search for tumor-derived chemoattractants, and this is one of the very early descriptions of a chemokine, MCP1 or CCL2. And in general, the picture that emerged from those studies and from studies, of course, by others, is that of a symbiotic relationship between macrophages and tumor cells, whereby tumor cells attract macrophages and macrophages serve as tumor cell little helpers. And this is single-cell analysis of macrophages done by Vasily Kandinsky. And you have the classically activated Alla Evans, Bob Evans, macrophages killing tumor cells, the alternative form of macrophage activation, and variations on the team. And I want to emphasize that we did the first, when we did, and this is Shubhra Biswas in the lab, who did the first profiling of tumor-associated macrophages, we found that things were more complicated than we believed. But essentially, there is still some merit in these simplified views. And in the last part of the talk, I will provide some real single-cell analysis of a novel lymphoid cell population involved in anti-tumor immunity. So macrophages have served as a paradigm for the connection between inflammation and cancer. They
the connection, we like to simplify things and think of the connection between inflammation and cancer. When I say we, it's here, we. Of the connection as consisting of two pathways, an extrinsic pathway whereby inflammatory condition, we mentioned, and we just discussed hepatocellular carcinoma, inflammatory conditions increase the risk of developing a tumor, and an intrinsic pathway whereby genetic events that cause cancer orchestrate the construction of an inflammatory microenvironment. And the inflammatory microenvironment include macrophages, neutrophils, which are dear to the heart of Andrea, and so the cellular arm of innate immunity, but complement. And the molecule that we cloned, this was actually our handle here, and this is work that was done by Eduardo Bonavita in my lab. Components of the humoral arm of innate immunity are important drivers of tumor-promoting inflammation. And in general, we are all aware that we have moved from a cancer cell-centric view of neoplasia to one that encompasses the tumor microenvironment, tumor-promoting inflammation, and taming of adaptive immunity. Naturally, my tenet would be that there should be an arrow here linking these two, and tumor-promoting inflammation, taming adaptive immune responses and T-cell responses. So this is, again, a scheme of tumor-associated macrophages, the macrophage balance, their diversity. In progressing tumors, these cells are skewed in a pro-tumor function by signals coming from tumor cells, from stroma, from polarized T cells, and I included complement because of the work done by Eduardo. And this is from a review that is in press, in Animal Reviews of Pathology. And these cells mediate immunosuppression. They mediate, promote invasion and metastasis, are components of a niche for, quote, cancer stem cells, and they influence response to hormones and drugs. And I lived at a time when there was only one, and this is another view of the same scheme, and this is taken from an editorial that Dan Longo and I wrote in New England Journal last December, and this is just to emphasize again the same concept, and also emphasize, and this meeting is running in parallel with a meeting on lymphomas, that in lymphomas there is proof of concept by Advani et al., Ir Weissman work on CD47, that if you block a don't eat me signal, so you re-educate what I called the corrupted policeman, you can have anti-tumor activity in humans. And it is my tenet that this may be the dawn of a myeloid checkpoint blockade era. So I lived at a time when there was only one cytokine associated with innate immunity, inflammation, and macrophages, and so we got involved in IL-1, a complex family. We identified the type 2 receptor as a decoy, cloned one of the receptor antagonists, cloned this fringe member of the family, and then showed that MYD88 is the signaling key adapter in the Tor, in the TLR and Toll-like receptor pathway. So early on, we asked the question, is IL-1 promoting tumor growth? And this is not a recent experiment. This was done in the 90s, early 90s, and we found that IL-1 promoted tumor progression. These are some of the papers. These data were confirmed, extended, and Ronnie Apted put these data on a firm genetic basis by using IL-1 receptor 1 gene-targeted mice. And this is the Cantos study, Paul Riedke. This, in my view, is a human carcinogenesis study. These are more than 10,000 patients, cardiovascular problems, high frequency of obesity, smoking, and high C-reactive protein. Blocking IL-1 beta resulted in 4 years in a more than 50% reduction in the incidence and mortality from lung cancer. The underlying mechanism, and this is a recent review of ours, involves, we believe, recruitment of myeloid cells, including macrophages and neutrophils, myeloid cell-mediated suppression of adaptive immunity, and skewing of adaptive immune responses. And I think that these findings raise several issues. Some are obvious, combination with checkpoint blockade. Some are less
obvious interception trial for patients who are at high risk, I think. I don't know what you think about it, and I think there is a big issue here, convincing people to, not people, but anyway. So let me go back to the balance. I will now go back to the balance and to the interaction of myeloid cells, macrophages, with innate lymphoid cells. And I want to very briefly summarize one published story and then move to two unpublished stories. And the published story relates to this molecule, which we cloned. This is a fringe member of the IL-1 receptor family, IL-1R8. We cloned it many years ago. Marta Muzzo cloned it many years ago in my lab. We focused on this molecule because it looked as a fringe member of the family, a single Ig domain, reminiscent of a decoy receptor for IL-18, long cytoplasmic tail, 2 amino acid substitutions in the TIR domain, which to us suggested that it should not be a signaling member of the family. And we were obsessed with negative regulation and decoy receptors. And indeed, work done by Cecilia Garlanda in my lab, and by Shishao Li in the United States, by Hans-Joachim Anders in Germany, and others have shown that this molecule is recruited at signaling receptor complexes of the IL-1 and TLR family and disrupts the formation of a midosome, so a negative regulator. In addition, there has been a recent twist in this story because Marcel Noll, Claudia Noll, Charles Tinarello, and we are on the paper because we provided the tools, to be honest, but it's really their story. We showed that this molecule is part of the receptor complex, together with the 18 receptor chain, that recognizes a forgotten member of the IL-1 family called IL-37. And IL-37 has long been known to be immunosuppressive. So again, negative regulation of immunity and inflammation. And the more recent twist came from work that was done in my lab by Martina Molgo. It's published work. We had found early on that IL-1R8 is very high in NK cells. It is also present in some T cells, I should say. And Martina found that this molecule, as such, it dampens signaling and IL-1-driven differentiation and activation of NK cells. In addition, work done by Miller et al. Has shown that IL-37 is produced by T regulatory cells and inhibits NK cells. If we inhibit and block, and IL-18 is produced by macrophages. If we block IL-1R8, this unleashes NK cell-mediated protection against primary carcinogenesis. We heard about primary carcinogenesis, and metastasis in the liver and in lungs. Why liver and lungs? Liver and lungs because we believe, because there are plenty of NK cells to start with, at least anatomical size. So there is a yin-yang function of IL-1R8 as far as carcinogenesis is concerned. On the one hand, it is a negative regulator of tumor-promoting inflammation. There is a recent study by Federica Riva in my lab showing that this is a pathway regulating lymphoma genesis, and there is a lymphoma, and we have correlative evidence in diffuse large cell B-cell human lymphomas. And on the other hand, it serves as a checkpoint in NK cells. We have evidence for T cells. And the general message is that unleashed NK cells indeed can mediate resistance against solid tumors. There were previous doubts about that. At least at NK cell-rich anatomical sites, such as liver and lung. I will now move to the two unpublished stories. And this is a story that started many years ago, about 15 years ago, with a talented PhD student, Fernando Martinez, from Cuba. At the time, we did the first transcriptional profiling of human monocyte to macrophage differentiation and polarized activation into M1 and M2. And among the M2 associated genes, we found a molecule called MS4A4A. And we decided to invest in this molecule, and we have not published the work
a paper. We have not published a paper for more than 10 years on this molecule because we wanted to understand. Why did we get interested in MS4A4A? Well, first of all, because it is a tetraspan-like molecule, a member of a large family of molecules of unknown function. The one that all of us know is CD20, with obvious implications, pretty obvious. But it's really amazing how much we don't know. Was mentioned about the hidden phosphoprotein. Here, I mean, there is an ignorome, a large ignorome of molecules we don't know the function. So this molecule is indeed, and this is, I should mention that this is work done by Irene Mattiola. Massimo Locati used to be in my lab, and we keep working together and supervising this effort in the last 10 years. So it is restricted to cells of the monocyte macrophage lineage, very low in circulating monocytes, induced during differentiation macrophages. It is widely expressed in tissues, but only expressed in macrophages, and not all macrophages, and not in all tissues. And I could belabor on that. It is indeed expressed in some tumor-associated macrophages, as we had predicted. And what does it do? This is why we did not publish a paper. And so we went fishing for a partner. We found that this molecule interacts with a pattern recognition molecule, dectin-1, which is also induced by IL-4 and M2-like signals. And this molecule, the tetraspan MS4A4A, is essential for driving dectin-1 at rafts, at lipid rafts, which are areas where signaling occurs. And indeed, we have generated knockout mice, we have generated lineage-restricted knockout mice, and the knockout mice have a problem in dectin-1 signaling. And they have basically a phenotype. The mice, MS4A4A deficient, as well as dectin-1 deficient mice, are more susceptible to metastasis, and this phenotype is related to NK cells. I will not go into the detail of the story because I want to save time for the discussion and for the second story, but this is the general message. We identified a member of a family of molecules, of tetraspan molecules, whose function is essentially unknown. This molecule is restricted to cells of the monocyte macrophage lineage. It interacts with the pattern recognition molecule, dectin-1, and it is essential for signaling by dectin-1. We assume it is essential for resistance against fungi, but we are not, this is not our priority. Recognition of lichen expressing poorly metastatic tumor cells triggers activation of NK cells via INAM and cytokines, NK cell activating cytokines, and this drives a pathway of resistance to metastasis. So this is the current model that we have. Here we have carcinogenesis, the Bob Schreiber and Mark Smith elimination phase, polarized, driven by interferon gamma, M1 activation of macrophages, but still we see in the clinic primary tumors with no metastasis. They have M2 or M2-like skewed macrophages. The cell, if they recognize N-glycans, they trigger an NK cell-dependent pathway of resistance to metastasis. And the loss of this pathway is permissive for metastasis. So this is our current model. But this molecule has implications that go beyond tumors, and actually the genetic evidence is striking for central nervous disease. And these are 17 papers in which if you dig into the actual data, you find very strong association between this genetic association and degenerative diseases of the central nervous system. And of course, again, this is a molecule that we really didn't know what it does. This is just for me to check my time. I think, Andrea, I have time, right? Yeah. And so I want to share you a second story. A second story that stems from my my preference for genetic data. Neutrophils are, I've been told by Gioacchino Natoli that neutrophils, in terms of epigenetic landscape, are failed monocytes. And the evidence showing related to the role of neutrophils in carcinogenesis is essentially dependent upon antibody-mediated depletion. And I like genetic evidence. And also there are limitations to the usage of antibodies because the anti-LY6G antibodies do not work very well in some mouse strains. They do not work very well in tumor-bearing hosts. And in addition, LY6G is also expressed on cells other than neutrophils. So we wanted to have genetic evidence. So we decided to use mice deficient for the growth factor of neutrophils, the G-Csf receptor knockout mice. And this is work that was done by Andrea Ponzetta, Sébastien Jaillon, and other people in the lab. And my prediction was that these mice, sorry, I forgot to mention, have a profound deficiency in neutrophils, and this is circulation, but essentially there are very few neutrophils at the tumor site, so very few tumor-associated neutrophils. And my prediction would be that these mice would be protected against carcinogenesis, neutrophils being part of tumor-promoting inflammation. I should also mention that I like primary carcinogenesis, and so this is primary, classic methylcholanthrin carcinogenesis. And what we found was surprising and unexpected, namely neutrophil-deficient hosts develop quicker tumors that grow faster. And the phenotype is rescued by adoptive transfer of neutrophils, unexpected observation. I should mention and emphasize that this, because there are young people here, that we want reproducible data, and Andrea has repeated this carcinogenesis experiment 20 times. So we are pretty confident as to the data. So we looked, last time I said we have repeated 7 times, and then he corrected and said, Alberto, we have done the experiment 20 times. We looked at the tumor, and we looked at cytokines, and the one cytokine causes gamma interferon, and found that in a neutrophil-incompetent host, there was less gamma interferon. The phenotype was rescued by adoptive transfer of neutrophils. And of course, we looked at the cells expressing interferon gamma in a neutrophil-competent and incompetent host. And here again, another unexpected finding, totally unexpected finding. No difference in terms of interferon gamma production by conventional T cells, CD4, CD8 cells, NK cells, not shown here, but a 60% reduction in neutrophil-incompetent hosts, in interferon gamma production in unconventional CD4 negative, CD8 negative, alpha-beta T cells. And these cells are skewed in a type 1 immunity direction, high EOMs, high T-bet, and in the absence of neutrophils, there is a loss of this skewing and skewing in a type 3 immunity direction. We did bulk RNA-seq on these populations, and bulk RNA-seq, we got what we expected, that in the absence of neutrophils, we got IL-17 production, and we had less T-bet, less EOMs, and less granzyme B, and more. And these cells, I should say, and I should emphasize, they have very high IL-12 receptor level. So we did single-cell analysis of these unconventional alpha-beta T cells, and I think that this is the first single-cell analysis at all of unconventional double-negative T cells in tumors, and this is a real tumor. Carcinogenesis, and we found 12 clusters of cells, and two of the clusters, cluster 2 and cluster 4, are neutrophil dependent for the skewing in a type 1 direction. And they are not classical unconventional double negative T cells. They are not the mate cells, for instance, that who are here.
or the gamma delta cells. They are not. It's a different population, new population. So what is it? What are their characteristics? And this is, they are endowed with a repertoire of innate immunity receptor, LY489 and Y49G receptor, innate-like repertoire. We do not have genetic tools to deplete these cells and to formally demonstrate that they are downstream of the protection against carcinogenesis. So we did the old man experiment, namely adoptive transfer. And this is This is adoptive transfer. This is adoptive transfer. These are two sarcoma lines. One double negative alpha beta T cell per 5 tumor cells, 1 per 30 tumor cells, and remarkable, in my view, reduction in tumor growth, given the ratio that we used. So the key question is this relevant to human tumors? So we looked at, this is sarcomagenesis. So we looked at human sarcomas, which are a mixed bag of tumors, and this is undifferentiated pleomorphic sarcoma, as we would expect. This is DCGA, and this is our own case list. As you would expect, a type 1 response is associated with good prognosis. Interferon gamma is associated with good prognosis. G-Csf receptor, so neutrophil infiltration, is associated with better survival, and a neutrophil signature is associated with better survival, and a neutrophil signature is associated with more interferon gamma. So this is in silico data, but we like old-style data. This is counting, and this is institutional case list, 19 patients, counting neutrophils. Here they are, high and low, and high neutrophil, better survival. And interesting and amazing for me, sarcomas are a mixed bag of tumors, and this correlation holds true only in undifferentiated pleomorphic sarcoma, and pathologists have held that these human tumors are the human counterpart of MCA sarcomas, which is, for me, is amazing. Is this only true in sarcomas? Well, no. This is an old paper of ours, Sébastien Jaillon. And we had found that neutrophil infiltration in colorectal cancer was associated with better prognosis. And there are 5 similar studies. If you look carefully at an in silico analysis done by Jérôme Gallon, you find the same. And again, G-Csf receptor expression is associated with, and this is a public database, sorry. So this is pretty solid, 5 studies, independent studies, large studies. This is public database, G-Csf high, better prognosis, neutrophil signature, better, and gamma, as we know, better prognosis. So we think we have identified a novel pathway of anti-tumor resistance, which involved an interaction between, again, this is work of Andrea Ponzetta and Sébastien Jaillon, which involves neutrophils interacting with macrophages. I didn't have time to dwell into that. IL-12-driven differentiation and activation of an unconventional T-cell population endowed with a repertoire of innate immunity receptors, innate-like lymphoid cells. And this pathway is essential for resistance against MCA carcinogenesis and, we believe, by association, selected human tumors. These are neutrophil nuclei in a patient with an acute phase response. And this is the neutrophil alphabet. And of course, P for us stands for PTX3 because PTX3 is a molecule that we cloned and it is present in neutrophil granules. But the general message is never underestimate the power of a neutrophil. And I hope I did not forget to mention the people who have done the work, Irene Mattiola and Massimo Locati. Massimo co-supervised with me the project for the MS4A4A project. Andrea Ponzetta is here, and Sébastien Jaillon for the neutrophil unconventional T-cell project, and the group picture of us at work. These are the PhD students and postdocs, the senior people, and this is my contribution. And this is the fact.
So thank you. So we have time for a couple of questions. Carl.
Great stories.
Yeah.
So 2 quick questions. One is what is the repertoire of these alpha beta double negatives? I mean, is it germline or do they have rearrangements? And then secondly, I mean, do these cells make cytokines like IL-12 or IL-18?
Yeah. The first question, we don't know. We don't know whether they have an invariant beta T cells, whether they are clonal or, as I suspect, they are not. Using Monocle, we have done a pseudotime differentiation of the double negative T cells in the tumor, and that's as far as we have gone, but we have to do this work. That's the first question. The second question relates, they are uniquely sensitive to IL-12. They are triggered by IL-12 under conditions. We have, in a reductionist approach, we have set up an in vitro, you put a STING agonist of TLR9 agonist, and you have neutrophils amplify IL-12 production by macrophages, and these cells are skewed and produce gamma. And what is striking for me is that on day 10 after you apply the carcinogen, the vast majority of the cells that are there are these cells. They are not the conventional T cells.
A question there, and
So our group was one among the 5 that described the positive prognostic significance of neutrophils in human colorectal cancer. Actually, we also found in vitro studies that neutrophils are able to co-stimulate proliferation and effector function of conventional T cells when T cells are activated under suboptimal conditions. So I wonder whether in the neutrophils deficient mice, you may have observed a deficiency of conventional T cells in terms of absolute numbers, or maybe whether a suboptimal stimulation of conventional T cells in this condition may also result in a deficiency.
Yeah, I think it's a very, very good point. The evidence we have is that the phenotype in terms of T cells in a neutrophil deficient host is restricted to the unconventional T cells. This does not negate that in a different context these cells can interact with conventional T cells, of course, because the mechanism that we have identified, I didn't have time to dwell into that, involves IL-12. Of course, IL-12 will not be, will not be private. These cells have actually in the bulk RNA-seq, what was emerging was very high levels of the beta chain of the IL-12 receptor. So they are ready to respond to IL-12. So that is our interpretation of the data. But of course, in the human, we have no evidence that in the human it's actually the double negative. I mean, we like to think that. And if you think it over, there is something strange in colorectal. I mean, we know since the work of Jérôme that even in stable colorectal, you have a prognostic significance of T-cell infiltration. But checkpoint blockade does not work. So there must be something else. And we had a look at some data, and again, I don't have proof of that. My tenet is that people have simply dismissed the double negative. Because we all look at 4 and 8 and forget about those that don't fit. That's my tenet.
Okay, last question. There is one question here. Sorry.
Yeah, a quick comment and a question. In anti-CD20 therapy in lymphoma, we see that there was macrophage. Actually, this usually correlates with a better outcome.
Yeah.
But I wanted to get your feedback on post-chemotherapy in a number of lymphomas. The ratio lymphocyte over monocyte post-chemotherapy reflects somewhat surrogate marker of the immune status or pro-suppressive or pro-tumor growth environment. How do you read this, and what do you think of the strategy is to try to fight this, to prevent recurrence?
Yeah, well, again, we are not saying that neutrophils are good always and under any conditions. I mean, the prognostic data that I showed you are only related to those particular tumors and that particular more carcinogenesis model that we have. And we are very well aware of plenty of data showing that high neutrophil to lymphocyte ratio is associated with bad prognosis, presumably emergency hematopoiesis, and myeloid cells. I mean, Andrea knows much better than I do that suppress adaptive immunity. So I'm not saying that that does not exist. I think that for all these cells, there is a yin-yang relationship that's depending on context. In terms of targeting, as I said, the work that we have done, but the CD47 work, the clever one work, and preclinical, recent clinical cancer research paper by the Finnish group, by Sirpa Jalkanen. But we know that there is a phase 1 trial with some interesting results, and that's, again, a myeloid checkpoint. So I think I would bet on myeloid checkpoints, but that's a biased view.
Okay, thank you very much, Alberto. Now we have like 10 minutes break, and we have time to have like a coffee break, which is supported by IPSA. Second section of this special symposium, IPSA. And so it's a great honor for me and a privilege to introduce the next speaker, which doesn't need any introduction. The next speaker is Carl June, and we are really happy that he has accepted to give a second talk during this ICML meeting. So Carl is a professor of immunotherapy in the Department of Pathology at the University of Pennsylvania. Is well known, probably everyone knows, because of the discovery of the CAR T cell, the implementation of the methodology of the CAR T cell. So we are really happy that he has accepted this invitation. Looking forward to hear about new perspective on CAR T cell.
Well, thank you very much, Andrea, and it's a real privilege to be here and to learn about IPSA and also the ICML meeting, which has been just fabulous learning experience. And so I have these conflicts to disclose, which are relationship with Novartis, where they've licensed the technology, and then with Community Therapeutics. And I'll show some early studies, clinical studies we're doing there. So, you know, CAR T cells are being used now in many institutions. They actually started in HIV, and it's an example of cross-fertilization of science, studying one for one purpose and then how it can lead to benefits in another. So a lot of the immunology that we know of and are used in cancer immuno-oncology now, we know of because of studies done on AIDS patients. And I'll just say that what CAR T cells are is the first example of synthetic immunity, where we use gene transfer to make cells that don't exist in nature. And you know, I showed on the talk on Tuesday that can change not only the signaling of cells, make them bispecific, but it also can change their metabolism from glycolytic to oxidative, depending on how you want that. And they then survive differently and can be enhanced their survival in hostile tumor microenvironments. So CAR T cells, you know, didn't just happen overnight. The first bispecific T cell where an antibody was used to redirect the specificity instead of HLA
or MHC binding to a peptide, which is what the normal T cell receptor does, was by Zeligesher and Kuwana, both independently showed that you could make a T cell work in a way that it didn't require to have the MHC complex binding of the endogenous T cell receptor. But then they grafted that onto a T cell receptor signaling domain, the zeta chain, and then other groups, and this was initially, it's very interesting, the initial experiments then with CAR T cells were actually done in biotech. So Helene Finney did the first co-stimulatory domain with using 4-1BB, which activates TRAF2 at a UK company, and Margot Roberts did in Cell Genesis, first tested CD28. Now, they never went past testing this in vitro because in 1999, there were gene therapy disasters that led the biotech to quit the field. They basically said it will never be possible to get commercial approval of gene-modified cells, or for that matter, adenoviral vectors even. So the biotech left, and the research stopped, and all then reverted to academics, who then took it forward. But it delayed the field by about 10 years. And the first people that actually did these experiments, as I mentioned, were women in biotech. So Helene Finney in UK, Celltech was called back then, showed that 4-1BB could make better signaling of the CAR when it bound its target molecule. Kristen Hagee, who I first began collaborating with in AIDS patients in 1997 with CAR-Ts, was then at Cell Genesis. She's now at Celgene and just had a paper in the New England Journal of Medicine showing that myeloma has very strong responses to CAR T cells targeting the molecule called BCMA. And so she stayed involved with CAR T cells and now in big pharma. And Margot Roberts was in Cell Genesis with Kristen and then went to Kite, which is now Gilead. And she made some of the first co-stimulatory TCRs. So the first CAR was actually this fusion protein of CD4. So CD4 normally, and Yvonne Horak, who's here, studied this a lot a long time ago when I was working with him in Bethesda. So this, it associates normally with the LCK protein tyrosine kinase, but Art Weiss grafted it on to the zeta chain of the TCR to study its signaling process. And they then retrospectively figured out that GP120 binds, the envelope protein of HIV binds very tightly to CD4, and that this then, if it's in a CD8 cell, could redirect the T cell to kill HIV-infected cells. So we did the first trials in HIV, and they were published in these trials in 2000, but we started in 97 testing that. And then, the FDA had a far-reaching rule, which was for anyone treated on a gene therapy protocol, you had to study them for 15 years to look for adverse effects. So unlike standard cancer therapy, we have a long database, and every year for 15 years, these patients came back. We published the results in 2012, finding, quite surprisingly, that the CAR T cells in a total of 40 patients had an average survival of greater than 17 years. So more than a decade survival, that's the half-life. And we just had 4 patients come back who were infused once in 1997, and they still have about 0.5% of all their T cells are CAR cells. They're CD8 cells that have a CD4 zeta in them. So that gives you now a long-term safety effect because not one of those patients has had side effects. And we don't ever have a database like that in cancer, because the FDA has not mandated, follow-up of long-term cancer treatments. And this is the first CAR trial done in cancer patients. And again, we did this with Cell Genesis in 1997. It was a colorectal cancer patients who had mets to the liver. Led at the clinical sites were UCSF and Stanford. About 15 patients were treated with 3 infusions of 10 to the 10th, so 10 billion CAR cells. It had a molecule that Jeff Schlom had made, an antibody targeting 72KD glycoprotein. So it was called CART72, and it was a first-generation CAR. It just had the zeta chain signaling, no co-stimulation like the current ones. And what we found was reasonable survival, but later the patients lost engraftment. So unlike our CD4 zeta, which is a fully human CAR, here they actually developed HAMR, antibodies to the mouse CAR, this this SCFV that came from a mouse hybridoma. So when you do solid tumors, you need to have fully human CARs, was one lesson. This was a patient with liver mets, were labeled with technetium scanning, which is all that they had back in 1997. And then a portion of the CAR cells were labeled here with indium-111, and they were injected in the portal vein of the patient who had a liver met. And what you can see is the liver met did not get any of the CAR T cells. So that's one of the central problems we have with CAR cells, is getting penetration of them into solid tumor masses. So this wasn't published until just last year. You know, we did the trial in 1997, and it just, because it was a failure and Cell Genesis went out of business, it was never published until retrospectively a year and a half ago. The initial clinical trials were done, the currently FDA-approved ones, all in academia. So the first trials of the CD72 was done by Kristen, and then at the NCI, Mike Kershaw and Steve Rosenberg treated patients with a folate receptor alpha CAR for ovarian cancer. And in the Netherlands, Cor Lammers treated patients with a GP targets carboxy anhydrase 9 CARs with kidney cancer. These didn't work at all. These first generation CARs that only have one signaling domain. So then the field and Jim Kotchendorfer treated the first patient at the NCI in 2010 who had a lymphoma, and then we reported our first 3 patients treated with leukemia in 2011. And these are what is now manufactured as by Kite Gilead as Yescarta, and then this is Kymriah that has a 4-1BB signaling domain. So our first patient was infused in 2010. He had end-stage chronic leukemia with a p53 deletion, which confers poor prognosis. We then have treated every B-cell malignancy that you can see listed here and had responses in all of them. And all of them it's an autologous T-cell engineered to express with a lentiviral vector, the CAR that I showed that has a 4-1BB signaling domain. But in all cases, it's an autologous T-cell. And the FDA approves the T-cell as a product. The drug is a T-cell. So it's different than a lot of other things where it would be, say, the vector or a small molecule. And then this shows, the design of the Yescarta and the Kymriah Novartis drug. The main difference is clinically, this one has persisted now we now know, more than 8 years on our first patients treated, and this one lasts on average 4 weeks. So there's a massive difference in the survival of the cells long term. And they have the same antibody binding CD19, but one's a lentiviral vector based, the other's a mouse gamma retrovirus, different promoters, different signaling domains, and we think that's the major difference here probably. But the response rate in lymphoma is the same. You don't need long-term engraftment in lymphoma, but in leukemia you do, unless you cross over to a secondary kind of treatment like a bone marrow transplant. So last year we published the 5-year survival data of our first 2 patients treated with the CD19 CAR who had had chronic leukemia. And David Porter's a physician, and Joost Mellenhorst did this. So the PK of the CARs is shown in red, and it goes out to 5 years on a semi-log plot. Basically, the CARs go up about 1,000-fold in the body, so there's massive proliferation. And then they collapse down to this so-called CAR memory level, which is a 2 to 3 log decrease in their frequency in the blood and bone marrow. And they remain functional, so we can show that this is the first living drug. If we re-isolate the CAR cells from the patient, they will still kill leukemia targets if the leukemia has CD19. And we found a number of molecular features of these CAR cells. They look like memory cells if you study memory T cells. Are CD27 positive, PD1 negative, CD8 positive, and they have an IL-6 STAT3 signature in them, which is different than most alpha beta T cells. So CAR T cells are really a different paradigm from the FDA's point of view. They're living drugs. They mature in patients and go through effector phases. And now the field's tested and treated more than 1,000 patients with CAR T cells, and not one has had the T cells get transformed. So they're very safe. There are plenty of results and reports where human stem cells after genetic manipulation have transformed and turned into leukemia. So stem cells have a different safety profile with genetic modification than T cells, and we could talk for a long time about why that is. It's an interesting issue in my lab. So CAR T cells are a process, and they're not just like a drug that you buy and give. So I mentioned they survive long term. The only thing in medicine that's really similar to this is a bone marrow transplant. And in that case, you find a donor, or if it's autologous, and get the cells out. And then instead of giving stem cells back, there's an in vitro manufacturing time, which defines what this vein-to-vein time is, how long it is from getting from the patient to returning them to the patient. And right now for Novartis, that's 22 days. It can be as short as 3 days with our current manufacturing, but right now for the commercial products, it's much longer. And so these are all autologous, the initial ones, but there's a lot of work on so-called third-party or off-the-shelf CAR cells where they're defined and derived by cells like iPSCs or cord blood T cells or healthy donor T cells. So, after we treated the CLL patients, we then began treating ALL, both in adults and children. And acute lymphoblastic lymphoma, this is a German multi-center safety and survival of 2,000 patients who had relapsed ALL. And the survival is just like it is for pancreatic cancer. It's about 10% at 2 years. And this is what has first become FDA approved in the US and now Europe, the treatment of multiply relapsed or primary refractory leukemia, and then now lymphomas. So there have been a number of side effects with CAR T cells. The most, and different from chemotherapy, they're on target in general. So that means it's a consequence of the CD19 molecule being targeted, either on malignant cells or on healthy B cells that have CD19. And that leads to cytokine release syndrome, which is now treated with tocilizumab, and I'm not going to discuss that. And high fevers. So cytokine release syndrome happens, but it's on target, and it only happens in the responding patients. Another, theoretic toxicity is, insertional oncogenesis, because, the DNA is broken when the lentivirus inserts its gene. So far, that has not caused any clinical events, and there's a lot of data that to say it's rare if it does occur. But what has been most vexing has been neurologic toxicity. None of our preclinical models predicted any of that, and about 1/3 of the patients treated with CD19 CAR cells have neurologic toxicity, usually after cytokine release syndrome, and it's usually entirely reversible, and it can be quite surprisingly focal. It's not a generalized encephalopathy. And the cause of that has been a complete mystery, and we have some recent data that's really interesting about that. So this is the first thing we found, this is our first leukemia patient, Emily Whitehead. When she was treated, she had a very severe cytokine release syndrome, and the PK, you can see the peak of her CAR T cells were about 10 days after infusion. And then at that time, 100% of her circulating cells were CAR T cells in the blood, and this is a Reichgemsa stain of what they look like in the peripheral blood. A hematopathologist would say it looks like an EBV infection with a lymphotropic virus. And surprisingly, they all get spinal taps at a month after treatment with the CARs, and we found all of our patients with ALL have CAR T cells in the spinal fluid. And even though they did not have any known CNS leukemia. So this is what if you take the cerebral spinal fluid and spin it down, that's a cytospin, that's what they look like. Those are CAR T cells. And so they go there, there's immunosurveillance there, and we think that may be part of the reason now of the neurologic syndrome. And this is very recent data from my lab at Penn and with Howard Chang's lab at Stanford, based on single-cell RNA-seq of the brain. So this is human formal brain, and what's done is the neurons are removed, and then the stroma is done single-cell mapping. And there's about 60,000 cells that was done. And they break down into the various cell types you would express. And parasites, which are the cells that line the blood vessels in the brain, have the dominant molecules you would express for a parasite like RGS5. But very surprisingly, they have transcripts for CD19. And CD19 is only been thought to be expressed in B cells. It's under PAX5 as a master transcription factor, and it's never been found in a non-B cell. So we found this in human brain and in mouse brain. And this just shows how they cluster out on Tisney plots. And the factors that you would expect to see in a parasite also is where you also see these CD19 transcripts. And this is by IHC staining. In human brain, these parasites express CD19. It's not diffuse throughout the brain. It's in certain anatomic lesions. And this is staining with a CD19 antibody. And cells that at least neuropathologists say these are parasites stained for CD19. And again, we see CD19 and expression at the transcript level, and then other genes that you would see in parasites. So it's expressed at the protein level. Avery Posey now was a postdoc in my lab and now has a lab pen of his own, has then made CARs targeting mouse CD19 or as a control human CD19 because it doesn't bind to mouse CD19, and then changed the signaling domains from 4-1BB or CD28. And what I didn't mention is that the clinical neurotoxicity is much worse with a CAR that has CD28 than if it has 4-1BB. And that has been unexplained as well. They have the same binder molecule on the outside of the T cell. So, and what we've done here is use Evans blue dye, EBD, and looked in immunocompetent mice or humanized immunodeficient mice and give them CARs, and then inject Evans blue dye a couple of days after the CAR T cells, and then sacrifice the mice or else do MRI of them to look for breakdown of the blood-brain barrier. As a positive control, we've used mannitol, which, this is an intact blood-brain barrier in a mouse, and this is one after mannitol where there's a lot of leak of Evans blue dye. And this is actually just looking here, and this is a CD28-based CD19 CAR. If it targets human CD19, it doesn't cause leak. So it's dependent on binding to CD19, and it's much worse with the CD28 signaling domain than it is with 4-1BB, which is here. So we think that this may be one of the mechanisms of the neurotoxicity with CD19 targeting. So there is, I mentioned some PK that's really different from a CAR than with standard drugs which go away and get metabolized. So, and this is compartmental analysis that was just published of CAR T cells, and we did this with 90 patients that we treated. They all had a CD19 CAR with a 4-1BB signaling domain. And we find that the CAR T cells expand with a rate constant of doubling of 0.78 days. So they're doubling probably every 6 hours, and some of those cells die, but the net accumulation is a 0.78 doubling time. Then they hit a peak at around, and that's a Cmax, at around 10 days. And then they have an initial alpha phase decay where the half-life is around 4 days. And we think this is associated when the target levels go down. So that if it's killing the tumor, then they go down. And then there's a beta decay rate, which in about 3/4 of the patients is essentially infinitely stable, and they have just long-term engraftment. But in some patients, they get immunized by the CAR, and then they reject the CAR, and that comes out, so an average of about 220 days. So that's the PK of these CARs that last a long time. This is just data showing, of 476 patients that we treated at the University of Pennsylvania, beginning with our CD4 zeta trials in HIV, which are shown here. And they're followed up every year. There we have over 780 years worth of patient safety. And all told here, 1,700, and this has not been updated for 2 years. So without any adverse effects. Now, if you give chemotherapy to people for breast or ovarian cancer and then wait, about 5% get leukemia 5 to 10 years later. So it's called secondary AML, and it's an awful disease. So chemotherapy is much more toxic, and that occurs at the stem cell level. Okay, so there are now emerging kinds of resistance to CAR T cells. And they're different for different tumors, even though they target CD19. So in the case of ALL, the main cause of resistance has been target loss, which means a subclone of the tumor comes back that no longer expresses CD19. And there's a pretty obvious cure for that, which is being tested at several institutions now where multiple CARs are infused at the same time, targeting other B lineage markers like CD20 or CD22. And this is ongoing at Stanford, Penn, many trials in China testing this. Now, Marco Ruella, who's here at this meeting, has shown that in rare cases, there can be an escape if the lentiviral vector transduces a leukemic cell as well as the T cells. And if the leukemic cell gets transduced, we call those CAR B cells. And then what happens is it masks the CD19 molecule, and even though the tumor cell is still expressing CD19, the CAR can't kill it. So the cure for that is improved tumor depletion before the CARs are manufactured. And then the final, an emerging issue really right now is the issue of T cell exhaustion. So T cell exhaustion is a state of epigenetic reprogramming of cells, and our genetic program changes, and they then basically don't do much anymore. They need to have chronic antigen stimulation, but they can no longer proliferate. It's different from senescence, and there's a lot of data written about what exhaustion is. We think it occurs quite commonly in the CAR T cells in chronic leukemia. And activation-induced cell death can also occur where the T cell gets stimulated too strongly and it actually then causes apoptosis. So Anjana Rao had a paper in Nature early this year looking at there's 3 genes in the NR4A family, and she showed that if she knocked those out, it prevented NFAT signaling, and it prevented the induction of exhaustion. And there were just 3 back-to-back papers came online in Nature this week showing a gene called TOX and TOX2. If you knock those out of T cells, then that prevents induction of exhaustion. And then there's this final one, which we published a paper last year in showing that TGF-beta signaling helps promote exhaustion in CAR T cells. So, and this is work now, the field's moving in solid tumors to so-called armored cars or trucks. Henrik Apken in Germany calls them trucks. Rainier Brenchens and Memorial Sloan Kettering calls them armored cars. But in solid tumors, the CARs have not had striking results like they have in bone marrow-derived tumors. And there's a lot of reasons for that: complex tumor microenvironment, genetic heterogeneity, and more checkpoints. Some ways around that are engineering the CAR T cells to express cytokines. Rainier Brenchens is presenting data on that today from the trial at Memorial Sloan Kettering. And we have a trial now with prostate cancer with our armored CAR, which is it has a dominant negative receptor to TGF-beta, so that it's insensitive then to TGF-beta, and targets the enzyme PSMA, prostate-specific membrane antigen, which is an ectoenzyme on, it can be in the stroma or on the carcinoma cells, which metabolizes glutamate into, basically it ends up in, you can get acetyl molecules, and then and actually that can feed the tumor microenvironment, so it helps the cells grow. So it's known in prostate cancer that the more advanced stage prostate cancer, you have more PSMA expressed, and it gets more brighter, basically. And it's also been shown in multiple ethnic groups that the more aggressive prostate cancers have more TGF-beta. And so for that reason, we made, and this is Chris Kloss and Jian Li in my lab made, with a lentiviral vector system, CARs that target PSMA, but that have dominant negative TGF-beta receptor. I won't go through all the data, but and it's published here. What this shows is an experiment where if you keep adding lots of prostate cancer cells to, and that's at every red arrow here out to 6 weeks, what you do then is that's a source of antigen, and CAR T cells then initially divide and then kill the tumor cells. And if they have intact TGF-beta signaling, they stop proliferating after about 21 to 28 days, and they're exhausted. And if you have them resistant to TGF-beta, they proliferate for at least 6 weeks, and there's less, they make more cytokines and less of the exhaustion genes that we showed in this paper. So that was the rationale for starting this trial. And the trial is ongoing at Penn now. It's a phase 1 trial led by Naomi Haas and Vivek Narayan at Penn. And it's a 3-cohort trial in patients with castrate-resistant metastatic prostate, initially giving 10 to the 7th cars per meter squared, and then 10 to the 8th per meter squared, and we've just started this with 2 patients now, where they're given fludarabine and Cytoxan, which is given as lymphodepletion, and then the CAR T cells. And so we have, this is from the first cohort of patients. Well, 2 out of 3 patients had good proliferation of these CAR cells. And this is the copies of the CAR per microgram genomic DNA. This is about 1.5 logs lower than what we see with CD19. And so we don't think we're getting high enough E to T ratio here, effector to target. But also here, the tumor is extravascular. It's not like leukemia, where they would stay in the bloodstream. So in a solid tumor, it's likely the CAR T cells have to leave the blood and go into the tumor, and we don't know those exit kinetics. So we found safety in cohort 1, some proliferation. Cohort 2, we had, for the first time, cytokine release syndrome in solid tumor patients. 2 of the 3 men required treatment with tocilizumab, which blocks IL-6 receptor signaling, and steroids, and they had decreases in their circulating tumor cells. And then we treated patient 1 of cohort 3, who, as shown here, he had exponentially increasing PSA for 4 months as a categorical axis. The CAR T cells were given here, and this is his PSA then. It went to unmeasurable levels within 21 days. PSA has a half-life in serum, according to Wikipedia, of 2 to 3 days. So this is equivalent to basically all the cells being killed within just a few days that made PSA following this. Now, he then had a very severe cytokine release syndrome, grade 4, which required steroids, tocilizumab, multiple doses, and he got, this is ferritin, is an inflammatory marker that you see goes up in cytokine release syndrome. So this is what we're seeing now, and this is, we think, is good. This is the proliferation of the CARs, again, the PK per microgram of genomic DNA. Cohort 1's in blue, cohort red is cohort 2, and then that's cohort 3 patient. So really much augmented proliferation, and that we've seen in other trials when you give lymphodepletive chemotherapy. This is the PK that I mentioned that has an initially 0.78 doubling time. Now we're seeing the same here as we saw there. So we're really excited that, you know, that's one thing, is the proliferation of the cells. So I'll just finish with this. This is from a Wells Fargo group showing CAR T cell companies, and there's over 300 now. When we started, it was just Novartis, and then Kite started. They're the only marketed ones. But if you go out on this diagram, it goes out to earlier and early stage cells, companies, and trials. So phase 1, phase 0 trials. And they're all these are CAR cells that are autologous. These are allogeneic CAR cells. These are T cell receptor modified cells. These are NK cells. This is where Alberto would be interested now. NK cells, NKT cells, and macrophages. There are CAR-Max even being tested. So it's really incredible, the diversity of development now, and it's happening much more rapidly than you could imagine. And it's not geographically uniform. If you notice on this heat map from clinicaltrials.gov, most of the development and trials are in the US and China. And Europe is a relatively cold region here. They have the same amount of preclinical development when you look at papers published, but the trials have been slower on the uptake here for probably geopolitical reasons. There's a lot of issues with the cost of cancer trials, and there's going to be a talk at the ICML that closed off the meeting today about this by Vincent Rajakumar, who's at Mayo Clinic. The average price of all drugs approved in the FDA in 2017 was $150,000 a year. So we have a problem that's generic to pricing of cancer drugs. One way to make CAR T cells cheaper is manufacturing improvements, and there's a lot of work on that. And this is data we published last year in Cancer Immunology Research. by changing the 10 to 12-day manufacturing down to 3 days. And in the immunodeficient mouse model, this is actually tumor bulk by bioluminescence. We actually found cells manufactured and using a subcurative dose of CAR cells, 500,000 per mouse, that this is what's marketed by Novartis. This is what we have now. They work better, and they're more potent on a per cell basis, and it costs a lot less to make them. It's 3 days instead of 12 days. So there's a lot of things going on in CAR T cell space these days, and I've mentioned some of these here. And you know what we're finding with CAR T cells is you only discover toxicity in phase 1 trials, and that's the same as a history of all cancer therapy. The preclinical models just do not tell you with any kind of precision what kind of toxicity to expect. This is my CAR lab that has done a lot of the experiments that I showed, and colleagues at the University of Pennsylvania and Children's Hospital. I highlighted the prostate cancer work being done, and the foundations that support our work. So I want to thank you for your attention.
Just a communication, since Solange Peters, next speaker, need to leave soon, can we wait for the question after Solange, the speaker? Okay, that would be great. I mean, it was a request. So it's a privilege for me to introduce the next speaker, which is Solange Peters. Solange is the head of the Medical Oncology Department at the Faculty of Biology and Medicine in Lausanne. She's been appointed as president-elected 2018-2019 ESMO president, and she's international known for the study, or better, for the application of immunotherapy to the field of lung cancer. So thank you for accepting our invitation.
Thanks a lot, and sorry for the change. Thank you, Carl, for moving the question to the end. I have to fly to Korea tonight, and if I miss the train, I will miss my flight. So yeah. I'm very sorry for inverting the thing, and very happy to come here and discuss with you the clinical part of the revolutions we have been experiencing in the lab, and you have been hearing too in the last 3 hours. I was asked by Franco Cavalli to cover a kind of a picture of the revolution in clinical applications today, which is probably not as revolutionary as you would expect for every of the aspects, but somehow we have changed the way we treat patients. My talk will focus quite a lot on solid tumors. We have been speaking about hemato-oncology a lot in this meeting, and I will also try to make a little experience for you in lung cancer, because that's my field of expertise, and maybe one of the solid tumors where this revolution took place already, which is a good example to guide the next solid tumors. This is my disclosure slide. So first of all, I'd like to keep in mind in whatever we do in oncology that applying precision medicine is a medical need, and it was defined as this model that proposes the customization of healthcare and all the aspects of healthcare with medical decision and practices being guided and tailored to each individual. I stress this one because sometimes when you make big discoveries, you forget about precision, and it's not because you have a new way to treat tumors, and I'll discuss it later on, that you should forget that every tumor is different. And if you look, it's a long way since this kind of nice picture of the karyotype of cancer cells, and this is a lung cancer cell line. You can see how obviously you might find genetic, very impacting differences between a normal cell and a cancer cell. And I like this paper, which is a kind of a landmark paper from Fogelstein and colleagues in Science, telling that by looking at 20,000 protein coding genes in many tumors, he was able to find an amazing number of mutations, 290,000 mutations, and by making a simple classifier, by looking at the redundancy of missense mutation and the redundancy of silencing mutation, classifying these mutations between tumor oncogene or tumor suppressor genes, he was kind of disappointing to find that only 125 drivers were found, 2/3 of them being tumor suppressor genes, genes. And however, it's already a lot because you can try to act on them in the clinic. But I also like this last sentence of this paper telling that we believe that the plateau is being reached because the same driver genes keep being rediscovered in different tumor types. And that's still what we experience. What we have been doing in lung is also being applied more and more in other solid tumors. Doesn't mean it's not good news to have these drivers, but it just means that probably we have taken the low-hanging fruits there, and we need to develop new strategies. But this is still something. And he also made this very nice graph classifying, and this biology for clinicians, classifying the fact that this driver will be a kind of concentrated and affecting 12 selected pathways that can be nominated as conferring a selective growth advantage to cancer cells within the carcinogenesis pathway. So in clinic, what does it mean? It means that if you have a mutation and a driver, this will result in the cancer phenotype. And we have in lung cancer very, very nice experiences with these drivers. And this is a very nice mice model of an inducible HER2 mutated protein, the same mutation which can be found also in other tumors, like, for example, breast cancer. And when you induce the expression of this protein in the lung of mice, you can see that it's necessary and sufficient to induce the development and the maintenance of several adenosquamous tumors of these mice. Really the driver definition. The problem of the driver definition is not always so pure. Sometimes the driver is not enough to really induce the malignant phenotype. And that's why with ESMO we made an attempt to try to put a context around the molecular tumor board. I'm sure in your institution you have a molecular tumor board when you will find this super rare mutation in a disease entity where it was never described, what to do with this mutation. And the problem is to try to classify a mutation by the level of evidence it gives you to propose a treatment to your patient. You need to be convinced to propose a patient. And this SCAT, which is a scale for ranking genomic alteration in the way and the magnitudes they can be actioned in terms of evidence-based medicine, goes from 1 to 5. And as you can see, 5 is an evidence-based, like a randomized clinical trial showing that it is a target in lung cancer or in colorectal cancer. And you will go up to 5 or even 6, where it's by principle potentially actionable, but nobody has ever shown that it is the case. And when you apply a 6 at your molecular tumor board, you should be very careful about the fact that sometimes doing clinical research on a single patient is not very ethical. Just wanted to put this word of caution. But basically going back to lung cancer and trying to apply these drivers in lung cancer. Lung cancer, this is the classification we are using now. When you look at the light blue curve, this is what we call a metastatic patient, somebody with 1 bone met and 1 liver met. The 5-year survival is not very difficult to evaluate. It's 0%. That's the baseline. And as you can see, the curve goes down very fast. Remember, the TNM will define it based on the tumor spread and the lymph nodes. And this is 2,000 attempts to make the fate of lung cancer patients better, chemo versus chemo versus chemo versus chemo, with no difference. But as you remember, an overall survival in metastatic patient of less than a year, 8 months, and basically a 1-year survival of a third of this patient, and chemo is equal to chemo, disappointingly. So from there, where did we make a revolution? This is my chapter 1, the drivers. We have been subdividing this non-small cell lung cancer into a subset, a numerous subsets of adenocarcinoma, non-squamous non-small cell lung cancer, with lots of them being actionable. In Caucasian patients, I would say a third of patients harbor a target which is easily actionable with an SCAT scale number 1. In squamous cell carcinoma, we also have identified drivers, but weaker drivers. And I must say, in the SCAT scale, we are closer to a 3 or 4 score, and this is not applied in clinical routine. But if I speak about the adenocarcinoma, and you will tell how fast could you develop so many drugs, it's because the biology helps us. This is the human kinome. So numerous kinases we need to have functioning in our body to maintain the homeostasis. And within this kinases, many of them, the tyrosine kinases, have a strong level of homology. And we have been developing drugs inhibiting some of these tyrosine kinases. And by homology, very often they have been derived from their first target to a second, a third, and a fourth target, because these inhibitors can act more or less on many targets in the same time. So where did it lead us? It led us in the last 5 years into this adenocarcinoma pie, and you can see my small subset. And this is an updated slide after the ASCO meeting, all the drugs that you can prescribe instead of chemotherapy in every of these little slides. So it really means that you can potentially propose to your patient something which keeps chemotherapy not for never, but for a later stage if needed in these cancer patients. And just to show you what we love in oncology is a waterfall plot. Every line is a patient. How does the tumor shrink with this targeted therapy? This is EGFR mutated. This is ALK rearrangement. This is the ROS1 rearrangement. This is the MET exon 14. This is the RET rearrangement. This drug function in a manner which is amazingly efficient in shrinking the disease. We will never have this study being done again. It's a very nice retrospective series comparing what the fate of patients with a driver at the era of targeted therapy to the time where no treatment was available. This is not ethical to randomize this patient anymore for chemo, but basically you can see that probably drivers allow to triple the survival of patients in non-small cell lung cancer in general. Okay? Let me give you an example of pharma development, of industrial development of drugs in the field of oncogene addictions. That's my preferred field, the ALK rearranged non-small cell lung cancer. Just for you to keep in mind that ALK protein is usually not expressed in the lung. It is expressed upon a rearrangement, putting a strong promoter in front of the ALK gene, usually the EML4 protein, and leading to the expression, to a massive expression of ALK protein in the lung, which is a very strong driver. And as you can see, we had this mice model where the expression, the conditional expression of ALK protein in the lung is a disaster, creating numerous adenocarcinoma in this lung, showing how potent it can be in inducing lung cancer. It's a very strange paradox for clinicians because for the first time, remember my first patient was a soccer player of 16 years old. This can happen in very young patients, which we never have seen in lung cancer. And interestingly, it happens more often in some, I would say, subcategories of patients, in particular in women. However, this is basically the age of the patients. You can see it in purple. The age of the patient, with more than half of the patient being less than 50 years old, probably 65% of them less than 50 years old, and many of them, almost all, never smokers. So young woman is a characteristic of this patient. We were not used to that in the clinic. So we've been developing drugs. Crizotinib was developed as a MET inhibitor, but ended up as being an ALK inhibitor and comes back to MET now. Just my homology story, right? And as a MET inhibitor, as an ALK inhibitor story, crizotinib was better than chemo. Not surprising, that's what I have shown to you before. However, we had some problems because crizotinib was wonderful, but the wonder did last only 9 months. In median time, after 9 months, the disease, for reasons which were still to be explained, was developing again. So we have been explaining in many publications how the development happens, resistance happens. It can be secondary mutation, it can be bypass tracks. These are the two main options. And the third one is the brain. ALK disease goes to the brain in an incredible manner and with a propensity which is difficult to explain, and crizotinib doesn't go to the brain. So the first site of relapse in most of these patients is the brain. If you have untreated brain mets and you give crizotinib, 70% of the patient will progress in the brain. So that was the resistance mechanism and the pharmacological mechanism of resistance in the brain. So this is where we have been incredibly active in developing drugs. All the new compounds you can see here are in the market and have some common properties. When I come to the common properties, this is the example of lorlatinib. I could have taken any of these drugs. First property, it has a more potent ALK inhibition function, a better affinity for the target, allowing you to penetrate even big tumors in a better manner. So second thing, it has activity against secondary mutations, so a broader activity against mutated ALK protein. And third thing, it has a wonderful CNS penetration. This is the second and third generation of ALK inhibitors. And here are the publications. It was my pleasure to go to this whole development with Roche and the Chinese colleagues of alectinib, and alectinib was strongly superior to crizotinib, as you can see on this progression-free survival Kaplan-Meier curve. And of course, what we wanted to show also here is it's incredible when these potent drugs are really addressing all the tumor cells. We have been able to update the PFS at the ASCO meeting last year, and we have now a PFS of 35 months. You start alectinib, and 35 months later on, you still have alectinib being efficient in these patients. And interestingly, we have resistance to this new generation ALK-TKI, which is also defined by the new set of secondary mutations. And you're frustrated because after maybe 5 years or 4 years, your crizotinib doesn't work anymore. One of the very important secondary mutations to new generation TKI is this so-called G1202R, which prevents the binding of most of this new generation TKI. And the pharma industry has developed a drug which is able to bind on another nucleotide than this G20, another amino acid than this G1202R, the lorlatinib, and you can see again that you can recover a wonderful response. So now we have 3 generations of ALK-TKI. We have 7 years median survival in the most recent series of a patient who was supposed to live 7 months at the beginning of the story. I just would like to raise an interesting point when I was speaking about the low-hanging fruits. We have been recently exposed to the NTRK fusion story, not only in lung cancer, but across diseases. It's observed in many disease types, but in lung cancer, if it is observed, it's observed in 0.3% of our patient population. But as you can see with all the NTRK inhibitors, you can see TRK inhibitors, you can see wonderful responses. And we went with this waterfall plots to the regulatory authorities in Europe, and they told us that's fine. You will have to make a clinical trial and randomize entrectinib against platinum-based chemotherapy to tell us that it's better than chemotherapy. Okay, so you will have to screen 100,000 patients. You will have to spend $400 million to make a trial for which you already know the outcome. You perfectly know that entrectinib is better than platinum-based chemotherapy. So that's a problem I can address briefly with you with what we propose to EMA. It's an ongoing discussion which is difficult. We all have this NTRK patient treated with platinum-based chemotherapy in our databases because we all do NGS. So you have this data in-house. If we all compose together this real-world evidence about the PFS, OS, and response rate of platinum-based chemotherapy of this disease, you have your standard arm. This is real-world evidence. It costs probably some millions, but not 400 millions. But this is EMA position until now. The role of real-world evidence for regulators is not about lowering regulatory standards at marketing authorization, and it's not to replace randomized clinical trials. So we still have a way to go, and this is something which, in terms of ethics to the patients and best treatment care, questions me on a daily basis. But we have to keep in mind, this is my wonderful first ALK patient in my trial. He made a wonderful response, and unfortunately, after probably 4 years, he developed his relapse first in lymph nodes in the lung and then everywhere. All these TKIs, all this oncogene addiction pathway, at the end, will not prevent, even if they are very well actioned on, will not prevent the relapse. Inevitably, this patient relapse. And it's very frustrating. You have this 5 years patient being controlled, coming for the routine CT scan. You look at the CT scan, a bit bored, and suddenly you have liver mets. So there's something in the Darwinian selection of cancer cells which will always surprise us. This is never curative, never. So what can you do? Second revolution, I go to chapter 2, the immunotherapy for the clinician. So we all know that we have these 4 pillars of promise that we work on strongly, and Carl June spoke about it, trying to enhance the T-cell priming, to remove the co-inhibitory signals like PD-1, PD-L1, CTLA-4, to supply co-stimulatory signals or to condition the microenvironment. And beyond the CAR T-cell, which were described by Carl, most of these 4 pillars are being studied with drugs addressing these potential ways to optimize durable tumor control and potentially, who knows, to obtain or to get to cure of some diseases. But today, as a medical oncologist, we basically target 2 synapses, the priming phase CTLA-4, happening usually in the lymphoid organs, and the effector phase PD-1, PD-L1 interaction happening in the tumor. And you all know that these drugs have been now available in the market, 2 CTLA-4 inhibitors, and many anti-PD-1, anti-PD-L1 for I would say, strategic reason and economic reason. You also have to know that more than 60 anti-PD-1 and anti-PD-1 are being developed in developing countries in order to allow for a market, first of all, of low social economic countries, but also the market of biosimilars in the future in order to lower the cost of this strategy. More than 60 are being developed. I saw 10 in my lab, all very good compounds. So I must say it's interesting to see that it might become a little less expensive in the future. So why is a medical oncologist excited? This is pembrolizumab. I could have taken any of these drugs. It works in some patients in all solid tumors. In some patients in all solid tumors. So let's go to the next slide. Unfortunately, it works in all solid tumors, but in a real minority of the patients. I would say a kind of a median average of 20% of the patients. So of course it's exciting, but due to the cost and complexity, 20% response rate is not sufficient. I just wanted to tell you what we are applying today. If you don't apply precision medicine, I will show you all the trials comparing second-line chemo to checkpoint in solid tumors. So no frontline, second-line, unselected tumor types using checkpoints. So this is a melanoma. If you go in second line against chemotherapy, DTIC, you can see a little more response, a little better outcome leading to registration. However, still not completely amazing. And you'll see with the other tumors, this is bladder cancer, pembro versus chemo second line. This is non-small cell lung cancer, atezo, pembro, nivo versus chemo second line. Of course, it's better. But do you find this is called precision oncology? I don't. Do you find this is called revolution? Maybe not really. Continue with that. This is head and neck, nivo versus chemo, or pembro versus chemo. This is renal cell carcinoma, nivo versus everolimus. And last but not least, this is gastric, where nivo and pembro shows the same improvement, but still far from being the revolution we expect. But we have moved a little bit more to precision oncology. How did we do? This is a table that you can also take on the website afterwards, with all the indications being now evidence-based in terms of going frontline with immunotherapy. How did we manage to do that? Two things. First of all, biomarker selections, meaning that suddenly you dive again into precision oncology. You need some biomarkers. Second thing, combinations, trying to improve the outcome on the checkpoints by combining with another immunotherapy compound or with chemotherapy. And you can see that the first column of frontline strategy is or biomarker selected or a combination. And with this, you can grab a little bit more out of your immunotherapy. So how can we improve immunotherapy outcomes for the clinician? So first of all, defining biomarkers. It's important because you need to enrich the population of patients you treat to take the good patients for your strategy. But also because you have to avoid harm. Harm is toxicity related to the drugs you use. It's the financial burden, of course. It is a type of harm. But it's also what some people did call hyperprogressive disease, or however you want to call it. It's also because when you have a treatment option which is standard in a patient, and you give immunotherapy in the wrong patient, the standard would have been better. Meaning simply that if you make the wrong choice in terms of patients, you might also have made a very wrong treatment decision, making the prognosis worse in this patient. So we need biomarkers for that. And we've been developing biomarkers on the tumor side, PD-L1, tumor infiltrating lymphocyte, and TMB. I will focus a bit on that. We've been developing peripheral biomarkers, which are still in the research arena, and we try to describe the host the best we can in order to select the patient. It's also still in the research arena. So in terms of clinician, we use PD-L1. These are pictures taken from lung cancer, but basically PD-L1 can be assessed by various assays that at the end are similar in their ability to be predictive in specific tumor types. But it's a biological continuum, so whatever threshold you use with PD-L1, you will deprive some patient who might have needed the drug because he's just under the threshold. And last but not least, PD-L1 is an easy biomarker because you can measure it also on a cytology small samples. But this being said, in lung cancer, we were very happy to see this ROC curve, not the best ROC curves you have ever seen, but this shoulder at an expression of more than 50% of the tumor cells with PD-L1, letting us thinking that high PD-L1 was predictive and 50% a good threshold. But this is the melanoma ROC curve, And you don't need to be a specialist in statistics to see that the predictive ability of PD-L1 is not something you would take into account in melanoma. And this predictive ability is insufficient in many tumor types, like, for example, also a renal cancer as a very good negative example. Of course, the problem is also that probably this biomarker is dynamic and heterogeneous. So I prefer another biomarker, which is a matter of huge debate in the community, and is a surrogate of a surrogate of a surrogate, which is the presence of neoantigens, but this is tumor mutational burden. It's a really far away, short way to try to show that some tumor might be more prone to present good neoantigens to specific immune systems. And we count mutations to do so, and we classify diseases by the number of mutations, just using this short way and paradigm that it might be increasing the probability of having strong neoantigen in the tumor. It's really a short way. So one would have thought that it's too easy to be applied. Unfortunately, or fortunately, it looks like it can, and despite the limited kind of scientific, not rational, but scientific applicability of mutations per se, you can see here that across all tumor types, if you look at the highly mutated tumors, the highest 20% or 30%, it's predictive across all tumor types of survival of patients treated with immunotherapy. So everything is about the threshold, everything is about the strategy you use, but this is a biomarker not like PD-L1 that you can apply. You will tell me doing a panel of gene, 400 gene, 1.1 footprint on DNA, in all tumor types, it's not possible, I agree. In lung cancer, I don't have tissue in half of my patients. However, we've been moving quite a lot, and I'm happy to have participated in this effort to move to calculate this tumor board in the plasma. So in the blood sample of patients, you can have another proxy of the proxy of the proxy of neoantigens by calculating the number of mutations in the circulating DNA. And this has been validated with a decent correlation with what you can observe in a panel in the tissue, and on the second hand, what you can observe with whole exome sequencing. So really meaning that the future is TMB in the blood, and you could even see over time is this fluctuates or is reduced or is increased, which can be interesting. This is the biomarkers. The first way was to have good biomarkers, and we like this immunogram for the Netherlands colleagues, telling you that it was easy. At the time we had mutational load, we had PD-L1, and now every month a new series of potential biomarkers is coming, and we still have to exploit some very important one, the old days data of the presence of TILs, which is defining as a predictive biomarker for all tumor types, the host characteristic in terms of HLA heterozygosy, which is very important, but we never apply it in the clinic. Of course, the microbiome, the target of so many clinical trials nowadays, and all these genetics components, looking at drivers, but looking also in certain tumor types at some mutations which characterize a primary immunoresistance. You can think about LKB1, STK11, which was nicely described as the resistant genes in the ASCO meeting recently. So this is going to be a complex story, because if you look at my little composition here, if you just use 8 biomarkers, you already divide your patient population, 3 biomarkers, you divide your patient population into 8 subgroups. And if you'd like to take into account everything I have told to you, with 7 primary parameters, and taking into account you're never sure they are completely interdependent, but imagining they are interdependent, you already defined 128 subgroups for which you should choose your treatment paradigm. So you will have to define in my immunogram which are the ones you'd like to push in the future. Based on that, and with this biomarker, one just curve for lung cancer, PD-L1 more than 50%. I have shown the ROC curves, and you can see how much simply Pembro is better in PFS, this was the primary endpoint, but also in OS and also in response rate. And using that in the good patient, and you can just concentrate on the left-hand side upper curve, is more than 50% PD-1 patients treated with pembrolizumab frontline. 30% of them are alive at 5 years, 1 out of 3. Remember, in the past, it was 0. What can you do for the other 2/3 of patients who don't have high PD-L1 to date? So we always based on the backbone of PD-1, PD-L1. It's interesting that the known backbone for our strategies, and we based on the knowledge we had of how chemotherapy, it was not just opportunistic. Chemotherapy can enhance the immune system, creating a parameter, a context in which the cell death is particularly immunogenic. We call it the immunogenic cell death. So the combination was opportunistic, of course, but was also rationally based, and it's important to say. And we also tried to combine with VEGF inhibitors because we know how much VEGF can be immunosuppressive. So a little opportunistic, but still some opportunities to grab. And we did it. And this is the green curve is chemo pembro versus chemo in purple. And you can see this is overall survival. You almost double the overall survival. And you can do it, interestingly, suddenly independently of PD-L1. So in all of your patients. But unfortunately here, you leave the precision oncology field because you will apply it to all patients. And you will be interested that since we have this combination reimbursed, people don't test anymore for ALK, EGFR, or all the drivers I described because they just apply the same formula to all. So new data should not contradict precision oncology, and that's a very important message. And probably I like this data. I had the opportunity to present at the wonderful AACR meeting. It was tumor mutational burden in the blood, the highest force of the patient using a CTLA-4 anti-PD-L1 versus an anti-PD-L1 versus chemo. I just would like to show you the blue curve in terms of overall survival. At 2 years, half of the patients with high TMB treated with the dual inhibition are alive, which is doubling what you get with chemo. And I'm quite convinced that these patients will have a long-term outcome, which is better than chemo combined with checkpoint. So we have to go back to precision oncology now, but we have changed the fate of our patients. I just published this recently with Sloan Kettering colleagues, just showing to the clinicians you have many new treatment opportunities. Don't give any more platinum-based chemotherapy if you can. And you will have to decipher in the future what is the best, depending on long-term outcome. That's the only parameter, long-term outcome. And we build, you don't have to know it by heart, this algorithm for all of the clinicians every day to try to help them to know if you don't have a driver, and that's of key importance, what you should do according to PD-L1 and TMB. Okay? We have developing many strategies in terms of trying to build in a desert an effective immunity. This can be CAR T cell, for example, how you can increase the immune response, for example, the checkpoints, and how you can also work on the microenvironment, for example, the adenosine pathway. We are all testing them in the clinic, and you'll see data probably in the next 2 years in lung cancer to a very large extent. The last important thing, we had biomarkers, combinations. It's something we started to observe in melanoma. It's striking how much immunotherapy works better in early disease. It might be because of the immunogenicity of the tumor, but I think it's mainly because of the patient status too. And the immune system and the exhaustion of the T cells might be quite different in early disease as compared to late disease. We interestingly also have some parameters showing that the neoadjuvant strategy, giving immunotherapy before any intervention on the tumor, is particularly favorable. And I have 3 slides before ending for you about this. This is atezolizumab, 2 cycles, 6-week treatment in a patient who undergoes surgery. And you can see here, this is the pathological response, that all of the patients have cancer cells being dead in the surgical sample. And among these patients, 20 of them had less than 10% viable cells in the tumor, which you usually never observe. So this was the first interesting data telling that neoadjuvant might be good before surgery. I even like more this trial where they combine 3 cycles of chemo and immuno in patients with stage 3 disease, the worst of the worst. And just look at that. Major pathological response, less than 10% viable cells, 90%. Complete response, according to RECIST criteria, 6.5%, 72% responders. Complete pathological response. Usually it represents 5 to 10% of our patients, 5% with chemo only. 70%. So here we have an opportunity to change the early disease fate, which is probably the most important thing. We are also giving now immunotherapy after radiochemotherapy in early disease because we have been showing that after radiochemotherapy you improve survival by giving 1 year of durvalumab, a type of maintenance or consolidation treatment, which also changes the early disease. What is interesting in this model, by the way, is that by doing durvalumab in the stage 3 radical treatment, you will also prevent the emergence of distant brain mets, showing how much immunotherapy works everywhere. A last slide about cost. You can see here in green on the left-hand side the cost of cancer drugs in the US recently. You can see on the dark line is the cost of supportive care, which is going down as compared to the cost of cancer medicine, and the big list of new drugs accepted by the FDA recently in the US. And just to go to my checkpoint, this is the unique patients treated with PD-1 or PD-L1 in the US between 2014 and 2018. Just explaining too, like Carl was telling, that we are strongly working on that with WHO on models allowing it to be sustainable over time, which would not be the case, will not be the case, if we do not define these models for the future. And my take home is we have to select frontline treatment option in every cancer types. And precision oncology means biomarker. It doesn't mean you find the best combination. It means that you will guide your treatment according to your patient's characteristic. Long-term benefit requires adequate selections. That's the only way to stay alive. And accessibility and sustainability will be the issue of tomorrow or are the issue of today. We will have to refine the biomarker signature, not to have 128 subgroups, use rational combinations and not having to face the IDO story again, and more than everything, move to early disease. We will screen for lung cancer, so we have to move for early disease very soon in order to really cure the majority of our patients. And with this, I thank you for your kind attention.
Thank you so much. Exactly. So, I mean, we should. I think Solange is leaving, but we have time, like, for some question to Carl June, Alberto, and. Oh. Yeah, go ahead.
Carl?
Yeah.
You. One question related to the neurotoxicity. There are data suggesting that blocking IL-1 may be a strategy, and in terms of biology, one should say that there is IL-1 beta in the CNS to start with. It's already there and for some reason. So I wonder what are your thoughts about that?
I totally agree. I mean, there's anakinra, IL-1 receptor antagonist, which I think is a trap. So, I mean, it's highly likely, as you're saying, that IL-1 is related to much neuroinflammation. And then the issues are in neurotoxicity with CAR T cells is, the drug that you give, will it get into the CNS? So like tocilizumab as a full antibody does not penetrate very well. So steroids do. And dexamethasone, but you know that they don't stop the fully activated T cells in humans. Dexamethasone is not good. So I think a small molecular weight, highly permeable molecule that blocked the IL-1 pathway would be very useful. And I was interested to note your data about on cancer progression and how the IL-1 and IL-1R8 is required. So I think there's a lot of potential there to potentially improve the efficacy of CAR cells and immunotherapy, but also the therapeutic index, difference between toxicity and efficacy.
I also have a question regarding the phase 1 trial with the PSMA CAR T. I remember by discussing with the Mark Rubin 1 month ago. Mark is a very good pathology for prostate cancer. He told me that PSMA level are somehow heterogeneous in the tumor, like in prostate tumor. So have you looked at, or it happened to you to see that you are actually selecting for clone where PSMA is negative?
So we're getting biopsies 10 days where possible in the patients after infusion of the CAR. So we very much think that in some patients, there'll be target loss where they'll have prostate cancer that becomes PSMA negative. Mm-hmm. Now, and that, one of the main issues evolving with very potent androgen receptor antagonists now is the emergence of enolase positive, so-called neuroendocrine prostate cancer. So that is something we're going to watch for. So we, at baseline, have immunohistochemistry on our patients and know kind of like with HER2 expression, how bright it is and how much heterogeneity. So in the end, it would likely require targeting more than one molecule, unless a patient has all cells are positive, or there is a so-called cross-presentation and epitope spreading to where other targets, are targeted by the endogenous immune system. I think, unless we have a strategy that does that, it will be very difficult to cure any solid tumor. Yes, Andre?
Yeah, thank you. That was a wonderful overview. I was wondering if you can comment on the CAR-T in lymphoma and leukemia. The activity is very impressive, but the failures are usually early. And what can we do, and what's the best strategies to prime the T cells, even maybe harvest, or how we can measure the suppressive environment, and what's the best strategy to overcome that?
So in what kind of leukemia?
In lymphomas or leukemias. Lymphomas, for example.
Yeah. So it's been a puzzling thing to us. We have found some data in our CAR T-cell trials in the solid cancer patients, for instance, pancreatic cancer treated with a mesothelin CAR, that there is some epitope spreading. In our CD19 treating lymphomas and leukemia, so far we haven't been able to demonstrate that. Now, as you know, most leukemias have a fairly low mutation rate. They have a few gatekeeper mutations, but they're on the low end of the spectrum as far as so-called neoantigens, very rare. So, and then the other problem really with treating these advanced leukemia and lymphoma patients is, very heavy pretreatment, and fludarabine is commonly used, and it's very immunotoxic. And once humans lose their naive T cells, they don't come back. Unless they're young and still have a thymus. So that's a real problem in humans that we don't have with other therapies. Is that if you depend on a subsequent immune response, taking into account all the chemotherapy. So there's some data that's in children, for instance, who get very high dose chemotherapy, coming on to all kinds of trials. This is from Children's Hospital in Philadelphia. They've looked at their T cells and in great detail. And if you look by ultrastructural analysis, the mitochondria are in terrible shape in those T cells. There's many mutations in the cells, and their proliferative capacity has been reduced a lot. So they're immunodeficient before they get any immunotherapy. And so one thing would be what Solange just mentioned. If things are moved up front, when the immune system's intact, it would likely work better. So both because there's less heterogeneity in the tumor and the immune response is more potent. So I think, the other aspect around this, where the field's going to go, is especially in these advanced stage patients, will be the so-called third-party cells. Allogene is developing cells that are coming from healthy donors, and then at least they do have any previous chemotherapy.
We have another question.
So I have another question for the neurotoxicity. So it looks like different CAR-T products have very similar level of neurotoxicity. But then on the other hand, one of them was better able to penetrate into the CSF at least. So that would suggest that whether they penetrated the CSF or even broadly speaking, the blood-brain barrier doesn't have anything to do with the neurotoxicity. So then is it just a matter of whether some patients, because it's not like everybody have it, is it because some patients has the CD19 positive parasites and then the majority of others don't? And therefore they don't.
So it's complex, and there's several layers to your question. So it turns out, for just high dose IL-2, lots of data, it can induce encephalopathy, and part of that is thought to be so-called just capillary leak. And then I think like the IL-1 system, like Alberto was mentioning, gets activated, and then glial cells and so on, and it becomes a cascade. So there's a lot of data about that. So part of in the neurotoxicity of CAR T cells is undoubtedly related just to the huge amount of cytokines that come up. And then what I didn't mention that's really fascinating about the parasites is it's only the parasites in the brain that express CD19. If you look at the ones in your arm, they don't express CD19. So there's some neuro connection there that we don't understand. We don't know what the function of CD19, if any, in a parasite, what that is. We know what its function is in B cells, where it helps, receptor signaling. So the parasite role, I think, is involved most likely with CD19 CAR T cells, but BCMA CARs can have very significant and CNS neurotoxicity that looks identical to the CD19 CAR. So we've looked, parasites do not express BCMA, but other cells in the brain do. They're probably glial-derived. We don't yet know much about that, but there may be different mechanisms, like with a BCMA CAR causing neurotoxicity that may be phenocopy in some ways of what CD19 is, but different in the molecular event. So it's like a lot of things, I think it can be very complex. And in humans that are outbred, we now know why some people get autoimmune disease when you treat with checkpoint therapies and some don't. That's a big mystery. And yet, there's so much we don't know yet about this on the susceptibility of toxicities.
Will I have a chance to ask one more question? So this is for treatment purpose. So I have a young patient, 24 years old, who had relapsed after CAR T and actually had a brain mass, multiple brain masses. He got the CAR T that doesn't cross the blood-brain barrier. Would you recommend that this patient may be a reasonable candidate for the one that does cross the blood-brain barrier? He has passed through many other treatments. So
is it a lymphoma?
Yeah, lymphoma, yes, I'm sorry, yes. And so we're pretty much running out of options. He got all the things, auto, chemo, PD-1, just pretty much everything.
So we have had, with our 4-1BB CAR, we've had people who have CNS lymphoma observed responses when they've been given intravenously. And we're now, initially our trial in ALL excluded patients with overt CNS involvement, but now we're treating people with active CNS leukemia and seeing responses. And some of them dramatic. Like we had patients with optic nerve and they were blind, and then the nerve started working again. We've also, there's a trial, for lymphangitic, meningiocarcinomatosis at MD Anderson, where they're injecting TIL cells actually right into the intraventricularly. And so I think that would be an experimental approach that this kind of patient might benefit from, because we don't know how the efficiency is of the cells that get in and how that would compare to direct. In brain cancer right now, there's a lot of work at City of Hope Hospital in Los Angeles giving intraventricular or intracranial CAR T cells. And there's data that not surprising, that it helps work better. So, unfortunately, that's an experimental level right now. And I'm not sure of where there might be trials for this, like basically a CNS lymphoma.
Thank you.
So thank you very much. I think we have to close the symposium. Thank you, Carl, and thank you everyone for being here today and for this beautiful day.